The coral reef at midday appears almost impossibly bright, a mosaic of living architecture where sunlight penetrates through warm, clear water and scatters across towers of limestone built by generations of tiny coral animals. Fish drift between the branching corals in bursts of color, while sea fans sway slowly in the current like underwater forests responding to a distant wind. Beneath this surface spectacle lies a battlefield defined not by claws or teeth, but by chemistry. Every inch of reef space is contested territory. Corals deploy microscopic harpoons filled with venom to defend their colonies from encroaching neighbors. Sponges manufacture toxic compounds that discourage predators from grazing on their tissues. Sea anemones extend delicate tentacles armed with stinging cells capable of paralyzing unsuspecting prey that wanders too close. In this ecosystem, survival depends as much on biochemical innovation as it does on speed or strength. Along one section of reef wall where algae form a soft green carpet across the limestone, a small creature glides slowly across the surface. At first glance, it resembles an animated fragment of the reef itself, an impossibly colorful organism with rippling edges and delicate folds that shimmer in the sunlight. Its body displays brilliant bands of electric blue, glowing orange, and deep violet arranged in patterns so striking they appear almost artificial. This is a sea slug, more specifically a nudibranch, an animal that seems to defy expectations about what a marine predator should look like. It has no shell to protect it from attack. It moves slowly, almost leisurely, and its soft body appears completely exposed to the dangers of the reef. Yet despite this apparent vulnerability, very few predators dare attempt to eat it. The reason lies in a strategy that borders on biological theft. Many sea slugs survive not by producing their own defenses, but by stealing the weapons of other animals and repurposing them for their own protection. The process unfolds through feeding behavior so specialized that it resembles a form of chemical piracy. Certain nudibranch species actively seek out toxic prey such as sea anemones, hydroids, and other cnidarians whose tentacles contain stinging cells known as nematocysts. These microscopic capsules function as biological harpoons. When triggered, they fire a coiled thread tipped with venom, capable of injecting toxins into prey within fractions of a second. For most animals, these structures represent a lethal obstacle. For the sea slug, they represent an opportunity. The slug approaches a colony of hydroids growing like miniature trees across the reef rock. Each branch carries dozens of tiny polyps surrounded by delicate tentacles, all armed with nematocysts designed to capture plankton drifting through the water. The sea slug begins to feed by extending a specialized mouth structure known as a radula. This flexible ribbon of microscopic teeth scrapes the hydroid tissue from the substrate and guides it into the slug's digestive system. Ordinarily, the ingestion of nematocysts would trigger their discharge, injecting venom into the predator that consumed them. Yet within the body of the nudibranch, the process unfolds differently. Inside the digestive tract, specialized cells isolate the nematocysts before they have a chance to fire. The slug then transports these intact stinging capsules through internal canals to structures along its back known as serrata. These elongated appendages resemble small fingers or fronds that sway gently as the animal moves through the water. Within the tips of these serrata, the stolen nematocysts accumulate inside storage sacs where they remain functional. In essence, the sea slug has disarmed the weapons of its prey and redeployed them as part of its own defensive system. This remarkable process is known as kleptomaniac, a biological phenomenon in which an organism appropriates the stinging cells of another species. Through this strategy, the nudibranch transforms itself into a living arsenal assembled from the defenses of multiple prey organisms. Any predator that attempts to bite the slug encounters a sudden discharge of venomous harpoons embedded within its skin. Fish quickly learn that these vibrant creatures are not worth attacking. Even experienced hunters that regularly consume soft-bodied animals avoid the brightly colored slugs drifting across the reef. The vivid coloration of these sea slugs is not accidental decoration. It functions as a visual signal known as aposematism, a warning display that advertises the animal's toxicity to potential predators. In terrestrial environments, this strategy appears in poison dart frogs and brightly colored insects whose patterns broadcast their chemical defenses. On the coral reef, the same evolutionary logic applies. The neon colors of nudibranchs act as a public announcement that their bodies contain potent deterrents. Fish that have once attempted to consume a toxic slug and experienced the unpleasant consequences quickly learn to associate those colors with danger. Yet the sea slug's story of chemical theft does not end with stolen stinging cells. Some species extend the concept even further through a related process known as kleptoplasty. In these cases, the slug consumes algae and retains not only the plant cells, but the chloroplasts responsible for photosynthesis. These captured chloroplasts continue functioning within the slug's tissues, allowing the animal to harvest energy from sunlight, much like a plant. Through this extraordinary biological arrangement, the slug becomes partially solar-powered, gaining supplemental nutrition from light while continuing to feed on organic prey. The combination of these strategies transforms the nudibranch into one of the reef's most unusual survivors. Without armor, speed, or physical strength, it thrives through chemical ingenuity. The reef environment itself encourages this evolutionary path. Coral ecosystems contain an immense diversity of chemically defended organisms whose toxins evolve to discourage predators. For most animals, these compounds represent barriers to feeding. For the nudibranch, they represent resources waiting to be appropriated. A closer look at the reef reveals how this strategy shapes ecological interactions. Hydroid colonies spread across exposed rock surfaces, competing with algae and small corals for space. Their stinging tentacles deter many grazing animals that might otherwise consume them. When a nudibranch species specializing in hydroids enters the area, the balance begins to shift. By feeding on these colonies, the slug reduces hydroid growth, indirectly allowing other organisms to expand. At the same time, the slug accumulates the hydroid's defensive cells, becoming increasingly well protected against predation. This pattern illustrates a broader principle within coral reef ecosystems. Chemical defenses do not simply prevent predation. They create intricate networks of relationships in which specialized predators evolve mechanisms to exploit toxic prey. Nudibranches represent one of the most striking examples of this dynamic. Their bodies become repositories of the chemical tools produced by other organisms, turning the reef's defensive arsenal into their personal shield. Despite their slow movement, these sea slugs display surprisingly complex sensory abilities that guide them toward appropriate prey species. Chemical cues dissolved in the water allow them to detect the presence of specific hydroids or anemones, even when these organisms remain hidden within the reef structure. The slug's rhinophores, two horn-like sensory organs located near the head, function as sophisticated chemoreceptors. By sampling the surrounding water for molecular traces released by prey organisms, the slug navigates towards suitable feeding grounds with remarkable precision. Once a nudibranch begins feeding on a particular species, its coloration often mirrors the appearance of that prey. This resemblance sometimes provides additional camouflage when the slug rests among the same organisms it consumes. The combination of warning colors and environmental blending may seem contradictory, yet both strategies operate simultaneously depending on the viewing angle and the predator involved. Fish approaching from a distance see the bright coloration as a warning, while small invertebrates living among the hydroid colonies may overlook the slug entirely because it resembles part of the surrounding structure. Reproduction adds another layer of complexity to the nudibranch's life cycle. These animals are hermaphrodites, meaning each individual possesses both male and female reproductive organs. When two slugs encounter one another, they exchange sperm simultaneously, ensuring that both partners can later produce eggs. The resulting egg ribbons often appear as delicate spirals or coils attached to the reef surface, each containing hundreds or even thousands of developing embryos. These larvae eventually drift into the plankton before settling onto the reef, where they begin the search for suitable prey species that will supply their chemical defenses. Predators that ignore the nudibranch's warning signals sometimes learn their lesson quickly. When a fish attempts to swallow one of these slugs, the stolen nematocysts embedded within the serrata discharge upon contact. The resulting sting is not always lethal, but it produces enough discomfort to discourage further attacks. Some species also secrete additional toxins derived from their prey, creating a combined chemical barrier that reinforces their reputation as unpalatable targets. The ecological implications of this chemical theft extend beyond the survival of the individual slug. By transferring defensive structures from hydroids and anemones into its own tissues, the nudibranch effectively redistributes venom within the reef ecosystem. The weapons originally evolved for prey capture now serve as anti-predator defenses for an entirely different species. This repurposing illustrates how evolution can recycle biological tools in unexpected ways, creating networks of interaction that blur the boundaries between predator and prey. Observing a nudibranch in motion reveals the elegance of this adaptation. The animal glides slowly across the reef surface on a muscular foot, leaving behind a faint trail of mucus that helps it adhere to the substrate. Its serrata sway gently in the current, each one containing clusters of stolen nematocysts ready to fire if disturbed. The bright colors shimmer beneath the shifting sunlight, broadcasting a message to any potential predator scanning the reef for an easy meal. In a habitat dominated by fast swimmers and heavily armored creatures, the nudibranch represents an alternative path to survival. It abandons physical defenses entirely and instead builds its protection from the chemical weaponry of other organisms. Through feeding behavior that resembles theft at a microscopic level, it transforms itself into a living collection of borrowed defenses. The coral reef remains a place where the boundaries between predator and prey are constantly shifting. Chemical warfare, camouflage, and sensory specialization all shape the relationships that allow thousands of species to coexist within a relatively small area of ocean. Among these interactions, the sea slug stands out as one of the most unusual participants. Its survival depends not on overpowering its enemies, but on converting their own weapons into a shield. As currents continue to sweep across the reef wall, carrying plankton and nutrients through the coral structures, the nudibranch drifts slowly across the substrate in search of its next meal. Somewhere nearby, another hydroid colony waits, armed with stinging cells designed to defend against predators. For most animals, those defenses represent a warning to stay away. For the sea slug, they represent a valuable supply of ammunition waiting to be collected. Through this quiet process of chemical appropriation, the seemingly fragile creature becomes one of the most untouchable inhabitants of the reef. The shallow reef flat lies just beneath the tide line, a broad mosaic of coral rubble, sandy channels, and living coral heads rising like miniature mountain ranges from the sea floor. Sunlight pours through the surface in rippling patterns, scattering across shells and algae with shifting brilliance. In these shallow waters, life moves quickly. Small fish flash through the coral branches in tight schools, while crabs patrol the sand, searching for fragments of food carried in by the current. From a human perspective, the scene appears visually rich, even overwhelming, yet it represents only a fraction of the information actually present in the reef environment. Much of the ocean's visual complexity exists in wavelengths, polarizations, and subtle light patterns invisible to the human eye. Hidden within that unseen dimension of light lives a creature whose perception of the reef surpasses nearly every other animal on the planet. Inside a narrow burrow carved beneath a piece of coral rubble, a mantis shrimp waits. Its body remains concealed within the tunnel, but two stalked eyes extend outward like periscopes, rotating independently as they scan the surrounding reef. The eyes move with precise mechanical control, swiveling upward, downward, and sideways, while the rest of the animal remains perfectly still. Each eye appears bulbous and segmented, with a band of specialized receptors running horizontally across the middle. To an observer unfamiliar with the biology of these crustaceans, the eyes seem almost comical in their exaggerated mobility. Yet these structures represent one of the most complex visual systems ever produced by evolution. Most animals perceive the world using a relatively small number of color receptors in the retina. Humans rely on 3 types of photoreceptor cells, each tuned to detect a different portion of the visible spectrum corresponding roughly to red, green, and blue wavelengths. By comparing signals from these 3 receptors, the human brain constructs the vast range of colors perceived in everyday life. The mantis shrimp, however, operates on a radically different system. Instead of 3 color receptors, its eyes contain 16 distinct types of photoreceptive cells. Each receptor responds to a narrow range of wavelengths, extending across the visible spectrum and even into ultraviolet regions of light that humans cannot detect. The result is a visual palette that appears almost unimaginable from a human perspective. Where a person might see a simple gradient of color across the reef, the mantis shrimp perceives a layered mosaic of spectral information. Ultraviolet reflections bounce from the shells of small crustaceans. Subtle shifts in wavelength reveal patterns on coral surfaces invisible to other animals. Even the skin of reef fish may contain ultraviolet markings that signal species identity or reproductive condition. The mantis shrimp processes all of this information instantly, translating the reef into a multi-dimensional visual map where color conveys far more than aesthetic beauty. Yet color detection represents only one part of the mantis shrimp's extraordinary visual system. Equally remarkable is its ability to detect polarized light. Polarization occurs when light waves vibrate along a specific orientation rather than oscillating randomly in all directions. In aquatic environments, polarization patterns often arise when sunlight reflects off surfaces or passes through water at certain angles. Many marine animals possess limited polarization sensitivity, using it to enhance contrast or navigate through complex light environments. The mantis shrimp, however, pushes this ability into an entirely different realm. Within the mid-band of each eye lie specialized receptors capable of detecting both linear and circular polarization. Circular polarization is particularly unusual because it involves the rotation of the light wave itself as it travels through space. To human vision, this phenomenon remains completely invisible. Our eyes lack the structures necessary to detect such patterns, meaning an entire dimension of visual information passes through our environment unnoticed. For the mantis shrimp, however, circular polarization functions as a clear signal embedded within the light of the reef. The evolutionary advantages of this sensory capability become evident when observing the behavior of mantis shrimp in their natural habitat. Although many species live solitary lives within burrows scattered across the reef, they still need to communicate with potential mates, rivals, and neighbors, occupying nearby territories. Bright coloration alone may attract unwanted attention from predators such as groupers and octopuses that patrol the reef in search of vulnerable prey. Instead, mantis shrimp often rely on patterns of polarized light produced by specialized reflective structures embedded within their exoskeleton. When sunlight strikes these structures, it becomes polarized in precise orientations or circular rotations, detectable only by animals equipped with the appropriate visual receptors. To predators lacking polarization sensitivity, the mantis shrimp's body appears relatively ordinary, perhaps displaying muted colors that blend with the surrounding reef. To another mantis shrimp equipped with the same sensory equipment, however, the signals become vivid displays of communication. These hidden patterns convey information about species identity, reproductive readiness, and territorial status without broadcasting the message to every predator in the neighborhood. This secret visual language adds another layer of complexity to the reef ecosystem. While fish and other animals rely primarily on visible color patterns or behavioral displays to communicate, mantis shrimp exchange signals through a private channel hidden within the physics of light itself. The reef thus becomes a place where multiple communication systems operate simultaneously, some obvious to human observers and others unfolding entirely beyond our perception. The mantis shrimp's extraordinary vision also plays a crucial role in hunting. Despite their relatively small size, mantis shrimp rank among the most formidable predators in shallow reef environments. Some species possess specialized appendages capable of delivering explosive strikes that shatter shells or impale soft-bodied prey. These strikes occur so rapidly that the limb accelerates faster than a small-caliber bullet, creating cavitation bubbles that collapse with additional force upon impact. Such weapons require precise targeting, and that precision depends heavily on the mantis shrimp's advanced visual system. Camouflage remains one of the most common survival strategies among reef organisms. Crabs cover their bodies with fragments of algae or sponge to blend into the substrate. Flatfish bury themselves in sand while adjusting their skin color to match the surrounding sediment. Small fish adopt mottled patterns that break up their outlines against coral branches. For predators relying solely on standard color vision, these disguises can prove extremely effective. The mantis shrimp, however, sees through many of these illusions by analyzing aspects of light that camouflage rarely conceals. Polarization patterns provide one such advantage. Even when an animal perfectly matches the color and texture of its environment, the way its body reflects polarized light may differ from the surrounding substrate. The mantis shrimp's eyes detect these subtle discrepancies immediately. A crab hiding beneath algae may appear invisible to human divers, yet its shell still interacts with polarized light in a way that reveals its presence to the shrimp's specialized receptors. Ultraviolet sensitivity adds another layer of detection. Many marine organisms reflect ultraviolet wavelengths differently from the surfaces they attempt to mimic. The mantis shrimp's ability to perceive ultraviolet light allows it to identify prey that otherwise blends seamlessly into the reef background. By combining ultraviolet detection with polarization sensitivity and a wide range of color receptors, the mantis shrimp effectively analyzes the reef environment through multiple overlapping visual channels. The mechanics of this visual processing remain a subject of ongoing scientific investigation. Unlike humans, who rely on complex neural comparisons between a small number of photoreceptors, mantis shrimp appear to use a more direct system in which each receptor corresponds to a specific wavelength or polarization state. Rather than constructing color through elaborate brain calculations, the shrimp may identify signals almost immediately at the level of the eye itself. This arrangement allows for extremely rapid visual processing, an advantage when hunting prey that can vanish into crevices within fractions of a second. Observations of mantis shrimp hunting behavior reveal how these sensory capabilities translate into action. A shrimp emerges cautiously from its burrow, its eyes scanning the surrounding reef with continuous motion. Each eye rotates independently, allowing the animal to survey nearly the entire environment without moving its body. The shrimp remains partially hidden while gathering visual information from multiple directions simultaneously. Suddenly, the eyes lock onto a specific point within the rubble field. What appears to human observers as an ordinary patch of sand actually contains a small crab attempting to remain motionless. Its coloration matches the surrounding sediment almost perfectly. Yet the mantis shrimp detects the prey instantly, guided by subtle differences in ultraviolet reflection and polarization patterns. The shrimp edges forward slowly, calculating distance and angle, while maintaining visual contact through both independently moving eyes. When the strike occurs, it unfolds faster than human vision can easily track. The mantis shrimp's raptorial appendage snaps forward with explosive acceleration, striking the crab with immense force. The impact not only crushes the prey, but also generates a brief flash of light produced by the collapse of cavitation bubbles in the water. This phenomenon, known as sonoluminescence, adds an almost surreal dimension to the attack, as if the strike momentarily pierces reality itself with a burst of energy. The combination of extraordinary vision and powerful weaponry places mantis shrimp among the most efficient predators within their ecological niche. Yet their success does not rely solely on aggression. The burrows they construct serve as protective refuges where the shrimp can retreat from larger predators and molt safely as they grow. Molting represents a vulnerable period when the exoskeleton softens before hardening again, leaving the shrimp temporarily defenseless. During these times, the animal remains hidden deep within its burrow, relying on its visual system to monitor the outside world through narrow openings. Even within the darkness of the burrow, the mantis shrimp's eyes continue their constant movement, scanning the environment for signs of danger or opportunity. The reef beyond the entrance continues its endless cycle of interaction, with fish hunting smaller fish, invertebrates grazing on algae, and currents carrying nutrients across the coral structures. Through its remarkable sensory equipment, the mantis shrimp interprets this environment in ways that no human observer could replicate. For scientists studying animal perception, the mantis shrimp represents a reminder that human vision captures only a small slice of reality. The reef environment contains layers of information embedded in ultraviolet wavelengths and polarization patterns that remain completely invisible to us. Within that hidden world, the mantis shrimp navigates with precision, reading signals encoded within the light itself. As the sun continues its slow arc across the tropical sky, the reef shifts subtly in color and brightness. Shadows lengthen across coral heads, while currents stir clouds of plankton, drifting through the water column. The mantis shrimp watches from the entrance of its burrow, its eyes rotating gently as they process the invisible dimensions of light surrounding the reef. For this small predator, the ocean is not merely a landscape of shapes and colors. It is a complex visual universe where polarization, ultraviolet reflection, and spectral detail combine to reveal a reality far richer than human perception can ever fully grasp. The tropical shelf begins as a gentle slope of pale sand and scattered coral outcrops, a seemingly quiet environment where the rhythm of waves above translates into a constant slow movement of water across the seabed. Sunlight filters through the surface in wavering columns, illuminating patches of reef where fish drift between coral branches and crustaceans scuttle through shadows. At first glance, the ecosystem appears calm, almost uneventful. Yet beneath this surface stillness, the ocean is filled with signals invisible to the eye, tiny electrical whispers generated by every living organism. Muscles contract, nerves fire, hearts beat, and each of these biological processes releases faint electromagnetic traces into the surrounding seawater. To most animals, these signals are meaningless noise, but to one predator, they form an intricate map of life hidden beneath the sand. Moving silently along the seabed, the hammerhead shark enters this invisible landscape as if stepping into a world that only it can perceive. The body of a hammerhead is unmistakable, and its wide head, the cephalofoil, appears at first like a bizarre evolutionary accident. Stretching outward in a flattened bar with eyes positioned at the far tips, the structure resembles an oversized hammer drifting through the water. Yet this peculiar shape is not a decorative quirk of nature. It is a highly refined sensory platform, one of the most sophisticated biological detection systems in the marine world. Along the underside of the cephalofoil lie thousands of microscopic pores, known as the ampullae of Lorenzini. These jelly-filled canals connect directly to electroreceptor cells, capable of detecting electrical fields so faint they approach the limits of physics itself. Every twitch of muscle from a buried fish, every pulse of a hidden heart, and every nerve impulse traveling through a prey animal produces tiny electrical disturbances. In the open ocean, such signals fade quickly, but across the sandy seabed, they remain detectable long enough for a hunter equipped with the right sensory hardware to locate their source. For most sharks, the ampullae cluster around the snout in a relatively compact arrangement, creating a localized sensory field directly ahead of the animal. The hammerhead, however, spreads these receptors across the full width of its cephalofoil, effectively transforming its head into a broad electrosensory antenna. This expanded array allows the shark to sample electrical signals from a much wider area at once, producing something akin to stereo perception. Instead of detecting prey as a vague electrical presence somewhere ahead, the hammerhead compares the strength and timing of signals arriving at different points along its head. By processing these minute differences, the shark can determine the precise direction and depth of hidden prey, even when the animal lies completely concealed beneath layers of sand. The result is a sensory system capable of locating life that is otherwise invisible, silent, and motionless. As the hammerhead glides above the sea floor, its movement appears unhurried, but the shark is constantly gathering data. Water flows over the cephalofoil, and within the pores of the ampullae, electrical signals stimulate specialized receptor cells that convert faint voltage differences into neural impulses. These signals travel along nerves to the shark's brain, where they are integrated with information from vision, smell, and the lateral line system that detects vibrations in the water. The hammerhead's brain devotes a remarkable portion of its processing power to interpreting this sensory flood. While human perception depends primarily on sight, the hammerhead experiences the ocean through layers of information that reveal hidden activity everywhere around it. The seabed below contains a mosaic of life adapted to concealment. Flatfish lie buried with only their eyes protruding from the sand, their bodies camouflaged so perfectly that predators relying on vision alone would pass overhead without noticing them. Rays settle beneath the sediment, their broad bodies disappearing under a thin layer of sand, while their gills continue drawing water through hidden openings. Crabs and shrimp retreat into burrows, sealing themselves within tunnels that offer protection from roaming hunters. To the hammerhead, These defensive strategies provide only limited security. Beneath the sand, muscles still contract and nerves still fire, and each electrical impulse leaks faint energy into the surrounding water. A hammerhead approaching such an area begins to alter its behavior subtly. The shark lowers its head closer to the seabed, sweeping the cephalofoil back and forth in slow arcs that resemble the movement of a metal detector scanning for buried objects. Each pass allows the ampullae to sample electrical signals across the sand. At first, the sensory landscape appears quiet, but gradually a faint disturbance emerges, an irregular pulse barely distinguishable from the background noise of the ocean. The shark adjusts its path slightly, bringing the signal closer to the center of its detection field. As it does so, the electrical pattern becomes clearer. Somewhere beneath the sand, a stingray lies hidden, its body generating a steady rhythm of nerve impulses that ripple outward through the water. The hammerhead's wide head now becomes a precision targeting instrument. Because the receptors extend across such a broad span, the shark can determine which side of the cephalofoil receives the strongest signal. This allows it to triangulate the location of the buried ray with extraordinary accuracy. The shark circles once, its body banking gracefully while the cephalofoil continues to sample the electrical gradient emanating from the sand. Each movement refines the calculation until the shark knows the exact position of its concealed prey. Then the pace changes. The hammerhead dips downward and presses its head against the sand with sudden force, pinning the ray beneath the weight of its cephalofoil. The unusual shape of the head proves useful once again. Acting like a flattened lever, it holds the ray in place while the shark maneuvers its jaws into position. Rays defend themselves with a venomous spine located near the base of their tail, a weapon capable of injuring or even killing predators that attempt to seize them carelessly. The hammerhead's strategy reduces this risk. By immobilizing the ray under the cephalofoil, the shark prevents the tail from whipping upward into a defensive strike. Within seconds, the hammerhead bites downward, gripping the ray and lifting it from the sand. The encounter lasts only moments, yet it illustrates how anatomy, sensory perception, and behavior combine into a coordinated hunting system. What appears from the outside as a strange head shape is in reality the central component of a biological machine designed to locate and subdue hidden prey with remarkable efficiency. The evolutionary path that produced the cephalofoil is still a subject of scientific investigation, but evidence suggests that natural selection gradually favored wider head structures in ancestral hammerhead species. Fossil records and genetic studies indicate that earlier sharks possessed more modest head expansions, which over time became broader and more pronounced. Each incremental increase likely improved sensory resolution, allowing sharks with slightly wider heads to detect prey more accurately than their competitors. Over thousands of generations, these advantages accumulated until the modern hammerhead emerged with one of the most distinctive silhouettes in the ocean. The cephalofoil offers additional benefits beyond electroreception. The wide spacing of the eyes provides the shark with an expanded field of vision. Unlike many sharks whose eyes face forward from a narrow snout, the hammerhead's eyes sit at the far ends of the head, granting nearly panoramic visual coverage. This arrangement allows the shark to monitor a vast area around its body, reducing blind spots and increasing awareness of both prey and potential threats. While the eyes do not deliver perfect three-dimensional depth perception directly ahead, the combination of vision, electroreception, and motion sensing compensates for this limitation. Hydrodynamics also play a role. The flattened head functions as a stabilizing surface that helps the shark maneuver through the water with surprising agility. As currents flow across the cephalofoil, they generate lift, allowing the shark to execute tight turns and maintain balance during sudden directional changes. This stability becomes particularly valuable when pursuing agile prey or navigating complex reef environments where rapid adjustments are necessary. The ecosystem surrounding the hammerhead benefits indirectly from this specialized predator. By targeting species that hide within the sediment, hammerheads influence the population dynamics of bottom-dwelling organisms. Rays, flatfish, and crustaceans all occupy important roles in the benthic food web, feeding on smaller invertebrates and recycling organic material within the sand. Predation by hammerheads helps regulate these populations, preventing any single species from dominating the ecosystem. This balancing effect contributes to the overall stability of coastal habitats, where interactions between predators and prey maintain biodiversity. Hammerheads themselves gather in large schools during certain times of the year, an unusual behavior among sharks that are typically solitary hunters. In tropical regions, massive groups can assemble along seamounts and island slopes, forming swirling aggregations that drift slowly through the water column. Scientists believe these gatherings may serve multiple purposes, including mating opportunities, social interactions, and possibly enhanced navigation using environmental cues. Even within these schools, the cephalofoil remains constantly active, sampling the ocean for electrical signals while the sharks move as a coordinated formation. Juvenile hammerheads begin developing their sensory capabilities soon after birth. Young sharks inhabit shallow coastal nurseries where mangrove roots and seagrass beds provide shelter from larger predators. Within these protected habitats, the developing hammerheads practice the scanning movements that will later define their hunting style. Small crustaceans and buried fish become early training targets, allowing the juveniles to refine their ability to interpret electrical signals from the environment. As the sharks grow, their cephalofoils widen proportionally, expanding the reach of their sensory array and improving detection accuracy. The hammerhead's electroreceptive abilities extend beyond hunting. The ampullae can also detect weak electrical fields generated by the Earth itself. Some researchers propose that sharks may use this sensitivity as a navigational aid during long-distance migrations. Ocean currents flowing through the planet's magnetic field produce subtle electrical gradients that animals like hammerheads could potentially follow. By sensing these patterns, the sharks may orient themselves across vast stretches of ocean where visual landmarks are absent. From a human perspective, the cephalofoil often appears exaggerated, almost cartoonish in its proportions. Yet this impression reflects the limitations of human perception, rather than any flaw in the shark's design. The hammerhead inhabits a sensory universe shaped by signals we cannot detect. To the shark, the ocean is not simply a blue expanse filled with visible shapes. It is an electrical landscape where every creature reveals its presence through faint pulses and fluctuating currents. Within this invisible world, the hammerhead moves with quiet precision. The cephalofoil sweeps through the water like a scanning instrument, translating biological electricity into a continuous stream of information. Beneath the sand, hidden animals remain unaware that their bodies betray them with every heartbeat. The shark approaches, guided not by sight alone, but by a multi-dimensional perception that integrates electrical, chemical, and mechanical signals into a unified picture of its surroundings. The result is a predator perfectly tuned to exploit one of the ocean's most overlooked sources of information. The cephalofoil transforms scattered electrical whispers into clear signals that reveal the location of prey concealed beneath the seabed. What appears strange or excessive from a human viewpoint is in fact a masterpiece of evolutionary engineering. In the silent waters above the sandy shelf, the hammerhead shark drifts forward, its head extended like a living antenna, reading the electrical language of the sea with extraordinary precision. Far from the warm, shallow reefs, where sunlight illuminates every coral branch, the open ocean descends into a world where light fades quickly and vision becomes almost useless. Beyond the continental shelf, the seafloor drops away into deep water trenches, where the sun's energy disappears within a few hundred meters. Beneath that threshold lies an environment defined by darkness and pressure, a cold and immense landscape where survival depends on senses other than sight. In this dim realm, sound becomes the dominant currency of perception. Water transmits acoustic energy with remarkable efficiency, allowing vibrations to travel across kilometers of ocean with little loss of strength. Every movement, every heartbeat, every flick of a fin sends subtle waves of pressure through the surrounding water. For most marine animals, these vibrations provide basic information about nearby activity. For one species, however, sound becomes a weapon of extraordinary power. The surface of the ocean may appear calm, but below it a massive shadow rises slowly through the water column. The creature approaches the surface in a controlled ascent, its enormous body drifting upward from depths where few other mammals can survive. When it finally breaks the surface, a burst of vapor erupts from a blowhole positioned slightly to the left side of its head. The exhalation forms a distinctive angled plume, a signature sign of the sperm whale. With a body length that can exceed that of many buses and a head that comprises nearly a third of its total mass, the sperm whale stands as the largest predator with teeth ever to inhabit the Earth's oceans. The whale's head dominates its silhouette, forming a vast rectangular structure that seems almost disproportionate to the rest of its body. Yet this massive structure is not simply an oversized skull housing a large brain. Inside lies one of the most sophisticated acoustic systems in the natural world. The sperm whale hunts primarily in the deep ocean where sunlight never penetrates, relying on sound rather than vision to locate prey. Over millions of years, evolution has transformed its head into a biological instrument capable of generating and focusing powerful bursts of sound with remarkable precision. At the front of this system lies the spermaceti organ, a massive chamber filled with waxy oil that occupies much of the whale's head. Above and around it sits another structure known as the junk, composed of compartments separated by fibrous tissue. Together, these tissues form a complex acoustic lens that shapes and directs sound waves produced inside the whale's nasal passages. When the whale prepares to emit a sonar pulse, muscular structures force air through specialized nasal sacs, producing a sharp clicking sound. Unlike the vocalizations of dolphins and many other toothed whales, these clicks are not merely simple noises. They are highly focused acoustic pulses, capable of traveling through vast distances of water. Once generated, the sound waves pass through the spermaceti organ, where their energy becomes concentrated and directed forward. The waxy oil inside the organ has acoustic properties that allow it to refract sound, much like glass refracts light. As the click moves through the various chambers and reflective surfaces within the whale's head, it becomes increasingly focused into a narrow beam, projecting outward into the darkness ahead. In effect, the sperm whale's head functions as a biological acoustic cannon, launching pulses of sound that illuminate the deep ocean through echoes rather than light. The intensity of these clicks ranks among the most powerful sounds produced by any living animal. Measurements recorded by marine researchers suggest that a sperm whale click may reach levels exceeding 200 decibels at its source. In water, where sound travels far more efficiently than in air, such energy carries across enormous distances. The returning echoes provide detailed information about objects encountered along the beam's path, allowing the whale to construct a three-dimensional acoustic image of its surroundings. As the whale begins its descent back into the depths, the ocean grows progressively darker. Within minutes, the sunlight fades to dim blue, then disappears entirely as the whale passes through the twilight zone of the ocean. Fish with glowing organs drift through the darkness, their bioluminescent patterns flickering like distant stars. Squid hover in the water column, their bodies nearly transparent except for large reflective eyes designed to capture the faintest traces of light from above. For these creatures, the deep sea represents a refuge from predators that rely on vision, yet the sperm whale navigates this environment using a sensory system that renders darkness irrelevant. Each click emitted by the whale expands outward through the water, traveling hundreds of meters until it encounters objects within the deep ocean. When the sound wave strikes a squid or fish, part of the energy reflects back toward the whale. These echoes return to specialized fat structures in the whale's lower jaw, which conduct the vibrations directly to the inner ear. By measuring the time delay and intensity of the returning echoes, the whale's brain reconstructs the location, size, and even texture of distant prey. Among the most formidable inhabitants of these depths are giant squid, elusive cephalopods whose bodies stretch across several meters of water and whose tentacles carry powerful suction cups lined with serrated rings. Encounters between sperm whales and giant squid represent one of the most dramatic predator-prey relationships in the ocean, yet they unfold in complete darkness far beyond the reach of cameras or human observation. Evidence of these battles appears on the bodies of whales that surface with circular scars etched into their skin, marks left by the squid's defensive tentacles. Before such confrontations occur, however, the whale may already have a decisive advantage. As the sperm whale closes in on a target detected through sonar, its clicking pattern changes. Slow, exploratory pulses shift into rapid bursts known as creaks, providing increasingly precise information about the prey's position. The acoustic beam narrows, concentrating energy directly toward the target. Scientists studying the physics of these clicks have proposed an extraordinary possibility. At extremely close range, the focused sound pulses may carry enough energy to physically affect the tissues of nearby animals. In water, intense sound waves can produce pressure fluctuations strong enough to disrupt delicate biological structures. For soft-bodied organisms such as squid, whose internal anatomy lacks rigid skeletal protection, the effect could be particularly dramatic. The acoustic energy may create vibrations within the animal's tissues, interfering with normal function and potentially causing internal damage before the whale ever touches its prey. Although the exact biological impact of these sonic pulses remains difficult to measure directly in the deep sea, the concept illustrates how the sperm whale's acoustic system serves multiple purposes. It functions as a navigation tool, a sensory probe, and potentially even a means of incapacitating prey through focused pressure waves. In essence, the whale hunts by projecting bursts of energy that reveal and possibly weaken its targets within the surrounding darkness. The whale's dive continues deeper, passing through layers of water where pressure increases rapidly. At depths approaching 1 kilometer, conditions become extreme. Temperatures hover near freezing, and the pressure exerted on the whale's body equals many times that experienced at the surface. Yet the sperm whale's physiology has evolved to handle these conditions with remarkable efficiency. Its lungs collapse safely under pressure, reducing the risk of nitrogen absorption that causes decompression sickness in human divers. Oxygen stored within the whale's muscles and blood supports extended dives that may last more than an hour, while the animal searches for prey. Within this silent abyss, the clicking rhythm continues. Each pulse travels outward like a probing finger of sound, touching distant objects and returning information to the whale's brain. The deep sea becomes a vast acoustic landscape where prey cannot hide behind darkness alone. A squid drifting hundreds of meters away may already have revealed its presence through the echoes bouncing back to the whale's lower jaw. Eventually, the whale detects a large returning signal, the acoustic signature of a substantial organism suspended in the water column. The clicking intensifies as the whale approaches. Somewhere ahead, a giant squid spreads its long feeding tentacles through the dark water, unaware that a predator is already mapping its position with increasing precision. The squid's enormous eyes, among the largest in the animal kingdom, search for the faint glow of approaching bioluminescence that might indicate danger. Yet sound arrives long before any visible cue. As the whale closes the distance, its sonar pulses accelerate into rapid sequences. Each click refines the target's position while the whale adjusts its trajectory. Within seconds, the predator reaches striking range. What follows remains hidden from direct observation, but the evidence left on the whale's skin suggests a brief yet violent struggle in the darkness. Tentacles lash outward, attempting to grip the whale's massive head, while serrated suction rings scrape across the thick skin. The whale counters with powerful jaws, lined with conical teeth designed to seize slippery prey. Moments later, the water settles again into silence. The whale swallows its prey and begins the long ascent toward the surface. Above, the ocean gradually brightens as faint light returns through the upper layers. The whale's lungs expand again as pressure decreases, preparing for the burst of air that will greet its return to the surface. This remarkable hunting strategy reveals how evolution has shaped the sperm whale into a master of acoustic perception. In an environment where vision fails completely, the whale transforms sound into a detailed map of the surrounding world. Its enormous head functions not as an awkward burden, but as a finely tuned instrument designed to generate and focus powerful sonar pulses. The deep ocean remains one of the least explored habitats on Earth, a vast realm where countless interactions occur beyond human sight. Yet through the biology of the sperm whale, glimpses of this hidden world become possible. Each click emitted by the whale carries information across the darkness, revealing prey that drifts silently in the abyss. The whale reads these echoes with astonishing accuracy, navigating through pressure and darkness, using a sensory system unlike anything humans possess. When the sperm whale finally surfaces once more, the ocean around it appears peaceful under the open sky. Waves roll gently across the surface, and seabirds circle overhead searching for fish near the waterline. Beneath this tranquil scene lies a different reality. one where sound replaces light and predators wield acoustic power as their primary tool. In that hidden realm, the sperm whale reigns as a giant equipped with a biological sonic cannon, projecting pulses of energy through the depths to locate, pursue, and ultimately capture the elusive creatures that inhabit the darkest corners of the sea. The tidal flats of the tropical Indo-Pacific appear deceptively simple at first glance, a wide expanse of shallow water broken by patches of sand, mud, and scattered seagrass. Sunlight reaches the seabed easily here, revealing an environment where camouflage becomes the primary currency of survival. Small fish hover just above the substrate, while gobies dart between burrows excavated by shrimp. Crabs shuffle across the sediment, leaving delicate trails behind them like signatures written in sand. In this open landscape, there are few places to hide, and predators patrol constantly. Barracudas cruise through the channels beyond the flats, while groupers linger near coral outcrops, waiting for a moment of opportunity. For smaller animals, the difference between survival and sudden disappearance depends on how effectively they can disappear into the background. Beneath the rippling surface of the sand lies a creature that has turned disguise into something far more sophisticated than simple camouflage. At first, the seabed appears empty, nothing more than a patch of pale sediment stirred gently by passing currents. Then the sand shifts slightly, as though the bottom itself has begun to breathe. A soft shape rises slowly from the substrate, unfolding arms that had been hidden beneath the surface moments before. What emerges is an octopus, but not one that behaves like the familiar reef-dwelling species known for hiding among rocks and coral crevices. This animal inhabits open sand flats, where concealment requires a different strategy. It is the Mimic Octopus, a master of transformation whose survival depends not only on blending into the environment, but on becoming something entirely different. The Mimic Octopus lives in a habitat where traditional camouflage offers only limited protection. On rocky reefs, an octopus can match the colors and textures of its surroundings while retreating into crevices when threatened. On a flat seabed composed mostly of sand, however, there are few features to mimic. A predator scanning the area would quickly notice any unusual shape resting on the sediment. The Mimic Octopus responds to this challenge by transforming its body in ways that extend far beyond simple color change. It alters its posture, texture, movement patterns, and even the apparent structure of its body to impersonate entirely different species that predators recognize and avoid. The foundation of this remarkable ability lies within the octopus's skin and musculature. Like many cephalopods, the Mimic Octopus possesses specialized pigment cells called chromatophores embedded within layers of skin. Each chromatophore contains tiny sacs of pigment that expand or contract under muscular control. By adjusting thousands of these cells simultaneously, the octopus can shift its color patterns within fractions of a second, producing stripes, spots, or uniform shades that match the surrounding environment. Beneath these pigment cells lie additional layers known as iridophores and leucophores that reflect light in different ways, allowing the animal to control not only color, but also brightness and reflectivity. Yet coloration alone cannot explain the Mimic Octopus's extraordinary disguises. The true secret lies in its muscular hydrostat body plan. Unlike animals with rigid skeletons, the octopus relies on networks of muscles that allow its limbs and body to bend, twist, and stretch in nearly limitless configurations. Each arm contains hundreds of suckers capable of gripping surfaces independently, while muscular bundles allow the arms to flatten into ribbons or thicken into rope-like structures. Because the body lacks bones, the octopus can reshape itself continuously, turning a rounded form into a thin sheet, or extending its arms into long, snake-like appendages. This flexibility allows the Mimic Octopus to perform a series of impersonations that appear almost theatrical in their precision. When a predator approaches from above, the octopus may flatten its body against the seabed, while extending its arms outward in opposite directions. The arms ripple in slow waves, while the body adopts a pale pattern with darker stripes along the edges. In this posture, the octopus closely resembles a flatfish gliding across the sand. Flatfish are common inhabitants of these tidal flats, and predators accustomed to seeing them may overlook the disguised octopus entirely. In other situations, the octopus adopts a far more dramatic transformation. If confronted by certain predatory fish that avoid venomous animals, the mimic octopus extends two arms forward while burying the remaining limbs beneath the sand. The exposed arms stretch straight out and begin to undulate with slow, deliberate movements. Dark bands appear along their length as chromatophores adjust to create a striking pattern. The result is an astonishing imitation of a sea snake, a creature many reef predators instinctively avoid due to its potent venom. From a distance, the illusion appears convincing enough to discourage pursuit. Another transformation involves the posture and coloration of lionfish, venomous predators armed with long, radiating spines. The mimic octopus spreads its arms outward in a circular pattern while adjusting the skin pigments to produce alternating light and dark bands. The arms extend and stiffen to resemble the spines of the lionfish, creating a silhouette that suggests danger rather than vulnerability. For predators familiar with the painful consequences of encountering lionfish, the resemblance may provide enough uncertainty to abandon the chase. What makes these disguises particularly remarkable is the apparent selectivity with which the octopus deploys them. Observations from marine biologists studying these animals in the wild suggest that the mimic octopus chooses specific impersonations depending on the type of predator encountered. For instance, when threatened by damselfish or other territorial species that aggressively defend their nests, the octopus may adopt the flatfish disguise and glide away along the seabed. When confronted by larger predators that fear venomous animals, the sea snake or lionfish impersonation becomes more effective. In this sense, the octopus does not simply hide. It actively analyzes the situation and selects the most convincing illusion for the moment. The tidal flats themselves contribute to the success of this strategy. These environments contain a wide diversity of animals with distinct body shapes and movement patterns. Flatfish slide along the bottom with subtle undulations, leaving faint trails in the sand. Sea snakes swim through the shallows with slow, sinuous waves. Lionfish hover among coral heads with their spines extended like delicate fans. Predators navigating this habitat learn to associate these shapes and movements with specific risks or opportunities. By reproducing those signals convincingly, the mimic octopus inserts itself into the predator's expectations and exploits them. The octopus's nervous system plays a crucial role in coordinating these complex transformations. Cephalopods possess some of the most sophisticated neural networks among invertebrates, with a large portion of their neurons distributed throughout the arms rather than centralized in the brain. Each arm can perform intricate movements independently while still responding to overall behavioral commands. This decentralized control allows the mimic octopus to adjust posture and movement patterns rapidly as conditions change. A passing predator may trigger a shift from one disguise to another within seconds, the arms reshaping themselves into entirely different forms. Beyond predator avoidance, these disguises may also assist in hunting. The mimic octopus feeds primarily on small fish, crabs, and other invertebrates that inhabit the sandy flats. By adopting the appearance of harmless species, the octopus may approach prey without triggering immediate escape responses. A crab that ignores a passing flatfish might react very differently if it recognized the approach of an octopus. Through mimicry, the predator effectively lowers the prey's guard long enough to close the distance. The interaction between mimicry and behavior reveals a deeper evolutionary story about the pressures shaping life on open seabeds. Unlike coral reefs filled with hiding places, tidal flats demand strategies that function in exposed environments. Animals must either move quickly, bury themselves in sediment, or deceive predators through disguise. The mimic octopus represents one of the most elaborate examples of deception in the marine world, transforming its body into a flexible canvas upon which multiple identities can be painted. This ability also reflects the octopus's remarkable capacity for learning and adaptation. Individual octopuses often refine their disguises over time, responding to repeated encounters with predators and adjusting their behavior accordingly. Such flexibility suggests that the mimicry is not purely instinctive, but influenced by experience. The octopus observes its environment through keen vision and tactile exploration, gathering information about the animals that share its habitat and the threats they represent. From a human perspective, the mimic octopus appears almost theatrical, performing elaborate transformations that resemble scenes from a stage production. Yet within the context of the tidal flat ecosystem, these displays represent practical solutions to constant danger. Every predator scanning the seabed carries its own expectations about the shapes and behaviors of potential prey. The octopus manipulates those expectations by presenting an image that contradicts what the predator hopes to find. As the tide begins to fall and the shallow water warms under the tropical sun, the mimic octopus returns to the safety of a burrow dug beneath the sand. The entrance remains barely visible, marked only by a small depression in the sediment. Nearby, gobies hover above their own burrows, while shrimp maintain tunnels that aerate the seabed. Life continues across the tidal flat, in a constant cycle of feeding and evasion. Somewhere above the sand, a predator passes by, scanning the bottom for movement. For a moment, the seabed appears empty. Yet beneath the surface lies a creature capable of becoming almost anything the situation demands. Through color, posture, and motion, the mimic octopus reshapes its identity in real time, transforming vulnerability into illusion. In the open landscape of the tidal flat, where hiding places are scarce and danger moves constantly through the water, this extraordinary shapeshifter survives by convincing the world that it is something else entirely. Along the shallow coastlines of tropical seas, mangrove forests extend their tangled roots into warm, sheltered water. These environments appear chaotic from above, a maze of arching roots and submerged branches that twist downward from the trees like wooden scaffolding. Sunlight filters through the leaves and reflects off the surface in fragmented patterns, scattering moving patches of brightness across the water. Beneath the surface, the world becomes quieter but far more intricate. Schools of juvenile fish weave carefully between the roots, using the labyrinth as protection from predators that patrol the outer reefs. Small crustaceans crawl across algae-covered bark, while tiny shrimp hover in the shadows, waiting for drifting particles of food. For many marine species, the mangrove forest functions as a nursery, a sheltered zone where the complexity of the environment offers both protection and abundant feeding opportunities. Yet this maze of roots also hosts one of the ocean's most extraordinary predators, a creature that appears almost too simple to navigate such complexity. Drifting through the filtered light is a box jellyfish, a nearly transparent organism whose delicate body pulses rhythmically as it moves through the water. At first glance, the animal resembles a floating cube of glass with long, trailing tentacles that extend downward like invisible threads. Unlike many other jellyfish that drift passively with the currents, this one actively swims, adjusting its direction and speed with purposeful contractions of its bell-shaped body. The water around it remains deceptively calm, but the presence of this creature introduces a level of danger that many animals in the mangrove forest instinctively recognize. The box jellyfish belongs to a group known as cubozoans, a lineage that diverged from other jellyfish long ago and developed features that seem almost improbable for such a delicate organism. Most jellyfish rely primarily on drifting motion, allowing ocean currents to determine their path while they capture prey that accidentally brushes against their tentacles. The box jellyfish operates differently. Its muscular bell contracts with surprising strength, producing directed pulses that propel the animal through the water in controlled bursts. This ability to swim with intention allows it to patrol specific hunting grounds, rather than simply drifting wherever currents carry it. Even more remarkable than its movement is the sensory system embedded within its transparent body. Suspended beneath the corners of the jellyfish's cube-shaped bell hang structures known as rhopalia. Each rhopalium functions as a sensory hub containing multiple types of light-detecting organs. Altogether, the jellyfish possesses 24 eyes arranged in clusters around its body. Some of these eyes detect simple changes in light intensity, while others possess lenses capable of forming images. This means the box jellyfish can perceive visual details within its environment, a trait rarely associated with animals lacking a centralized brain. The mangrove habitat makes such vision especially valuable. Sunlight filtering through the trees produces a patchwork of shadows that shift constantly with the movement of waves and leaves. Small fish seek refuge among the roots, darting between gaps in the wood as they search for plankton drifting through the water. For a predator navigating this environment, the ability to detect shapes and contrast becomes a critical advantage. The box jellyfish's image-forming eyes allow it to distinguish vertical structures, such as mangrove roots, and avoid collisions as it moves through the forest. Observations of these animals in the wild reveal that they actively steer around obstacles, adjusting their path to weave through the tangled underwater architecture. This navigation ability challenges the long-standing assumption that jellyfish operate as simple organisms guided purely by reflex. Despite lacking a central brain, the box jellyfish demonstrates behavior that appears surprisingly sophisticated. It can approach areas where prey is abundant, avoid shaded zones where its vision becomes less effective, and maintain distance from obstacles that could damage its fragile body. The explanation lies within a nervous system organized in a fundamentally different way from that of most animals. Instead of concentrating processing power within a single brain, the box jellyfish distributes its neural network throughout its body. Nerve rings encircle the base of the bell, connecting sensory organs with muscle tissues responsible for swimming. Each ropalium processes visual information locally, sending signals directly into the nerve ring, where they influence movement and tentacle responses. Rather than funneling all sensory input to a central command center, the jellyfish relies on a decentralized system in which multiple neural nodes cooperate to guide behavior. This distributed intelligence allows the animal to react quickly to changes in its environment. When the eyes detect a dark, vertical object that could represent a mangrove root, signals travel through nearby nerve pathways that alter the contraction pattern of the bell. The jellyfish shifts direction slightly, gliding around the obstacle before resuming its forward motion. At the same time, other sensory receptors monitor water movement and chemical cues, providing additional information about nearby prey. Trailing beneath the bell are long tentacles lined with thousands of microscopic stinging capsules known as nematocysts. These structures function as highly specialized harpoons capable of injecting venom into any organism that brushes against them. The venom of certain box jellyfish species ranks among the most potent in the animal kingdom, capable of immobilizing fish almost instantly. For the jellyfish, however, the venom serves not merely as a defense, but as an essential hunting tool. As the jellyfish drifts slowly between the mangrove roots, small fish move cautiously through the water column. Many of these fish have evolved keen senses for detecting predators, yet the near transparency of the jellyfish makes it difficult to spot against the shimmering background of filtered sunlight. When a fish ventures too close to the trailing tentacles, the nematocysts fire automatically, releasing microscopic threads that penetrate the fish's skin and deliver venom with astonishing speed. The prey's muscles contract involuntarily as the toxin interferes with nerve function, leaving the fish unable to escape. Unlike some jellyfish that rely purely on chance encounters, the box jellyfish appears capable of positioning itself strategically where prey activity is highest. Mangrove roots provide excellent hunting grounds because they attract dense concentrations of juvenile fish seeking shelter. The jellyfish swims slowly along these corridors, adjusting its orientation so that the tentacles spread behind it like an invisible net. As fish dart between the roots in search of food, they risk crossing the delicate threads trailing through the water. Once prey becomes ensnared, the tentacles contract gradually, drawing the immobilized fish toward the underside of the bell where the mouth is located. The jellyfish then folds its body slightly, guiding the captured prey into its digestive cavity. Within this internal chamber, enzymes begin breaking down tissues, allowing the jellyfish to absorb nutrients directly through its internal surfaces. The distributed nervous system continues to play a role even during feeding. Sensory receptors along the tentacles detect chemical signals released by struggling prey, reinforcing contraction patterns that bring the capture closer to the mouth. Meanwhile, the eyes remain active, monitoring the surrounding environment for obstacles or additional prey. This simultaneous processing of visual and tactile information allows the jellyfish to maintain navigation even while feeding. Researchers studying cubozoan behavior have observed that these jellyfish tend to remain within shallow coastal habitats where light conditions support their visual system. When water becomes too turbid or darkness increases beyond a certain threshold, the jellyfish alter their movement patterns, often rising toward brighter layers of the water column. Such behavior suggests that their vision is not merely incidental, but central to how they interact with their environment. The presence of multiple eye types also contributes to this flexibility. While the lens-based eyes detect shapes and edges, simpler light-sensitive organs measure overall brightness. Together, these sensors allow the jellyfish to gauge its position relative to the sunlit surface and the darker zones beneath mangrove roots. By maintaining orientation within this gradient of light, the animal optimizes its ability to detect prey while avoiding collisions with submerged structures. From an evolutionary perspective, the box jellyfish represents a remarkable experiment in alternative neural design. Many animals with complex behaviors rely on centralized brains that coordinate sensory input and motor responses. The cubozoan approach distributes that coordination across several interconnected nodes. Each sensory center contributes partial information, and the combined activity of these networks generates behavior that appears purposeful and adaptive. The mangrove forest provides a perfect stage to witness this system in action. Currents push gently through the roots, carrying plankton and small organisms that attract fish. The fish in turn attract predators, creating a layered food web that depends on the structural complexity of the habitat. Within this environment, the box jellyfish drifts with quiet precision, its transparent body almost invisible against the shifting light. The rhythmic pulsing of the bell continues as the animal glides past another root cluster. Sunlight flickers across its surface, illuminating the delicate geometry of its cube-shaped body. Beneath it, the tentacles extend like fine threads woven into the water itself. Every movement of the jellyfish reflects the coordination of countless nerve cells distributed throughout its body, responding collectively to visual cues, chemical signals, and the subtle mechanics of water flow. To an observer accustomed to associating intelligence with large brains and complex skeletons, the box jellyfish challenges familiar assumptions. Here is a creature with no centralized brain, no bones, and a body composed largely of water, yet it navigates a dense underwater forest with surprising competence. It detects obstacles, pursues prey, and adjusts its movement in response to environmental conditions. In the filtered sunlight beneath the mangrove canopy, the box jellyfish continues its silent patrol. Small fish glide cautiously between the roots, unaware that an almost invisible hunter moves among them with calculated precision. Through distributed perception and coordinated motion, this delicate predator transforms a seemingly simple body into a highly effective hunting machine. Within the quiet complexity of the mangrove forest, the box jellyfish demonstrates that intelligence in the natural world can emerge not only from centralized brains, but also from networks of sensing and response spread throughout an entire living body. As the tropical sun lowers toward the horizon, coral reefs undergo a transformation that reshapes the entire rhythm of life. During daylight hours, the reef exists in constant motion, illuminated by intense sunlight that reveals every crevice, coral tower, and drifting cloud of plankton. Fish patrol the reef slopes in colorful schools. Herbivores graze steadily across the limestone surface, and predators cruise through the open water searching for opportunities. Yet as evening approaches, the character of the ecosystem shifts dramatically. The water darkens slowly, and the activity of many daytime animals begins to fade. In their place, creatures of the night prepare to emerge. Among them are hunters that rely not on sight, but on scent. Animals capable of detecting faint chemical signals drifting through the water long after darkness has erased all visual clues. Within this daily transition, survival requires strategies that anticipate a completely different set of threats. Many fish that spend their days feeding openly across the reef now search for places to rest. Some wedge themselves into crevices between coral heads. Others bury themselves in sand where predators may struggle to locate them. But one group of reef inhabitants employs a solution so unusual that it initially seems almost impossible. Before sleeping, they wrap themselves inside a bubble made from their own mucus. The architects of this nightly cocoon are parrotfish, herbivorous reef grazers whose constant feeding plays a crucial role in shaping the coral ecosystem. During the daylight hours, parrotfish move methodically across the reef, scraping algae from coral surfaces with powerful beaks formed from tightly fused teeth. These teeth resemble the beak of a parrot, which gives the fish its name. but their function goes far beyond simple biting. The jaws of a parrotfish operate like biological grinders, capable of removing algae while also breaking down small pieces of coral rock. Inside the throat lie specialized grinding plates that crush these fragments into fine particles. Much of the coral sand found on tropical beaches originates from the digestive systems of parrotfish that consume limestone while grazing and later excrete it as fine sediment. In this way, the fish serve as both gardeners and geological engineers, continuously cleaning coral surfaces while contributing to the formation of reef sands. Throughout the day, these fish must remain vigilant. Their bright colors make them conspicuous against the reef backdrop, and numerous predators patrol the same environment. Reef sharks, groupers, and barracudas all regard parrotfish as potential prey. Yet daylight also offers the protection of visibility. A grazing parrotfish can detect approaching threats and retreat quickly into coral crevices if necessary. As long as the sun remains above the horizon, vision dominates the sensory landscape of the reef. Nightfall changes that balance entirely. When darkness spreads across the reef, visual cues disappear and many predators switch to alternative senses. Among the most formidable of these nocturnal hunters are Moray eels. Hidden within narrow crevices during the day, Morays become active after sunset, slipping out from their rocky dens to patrol the reef with slow, deliberate movements. Their elongated bodies slide through coral openings with ease, while their highly developed sense of smell guides them toward prey. Even the faintest trace of chemical scent drifting through the water can lead a Moray eel directly to a resting fish concealed within the reef structure. For most fish, this poses a serious challenge. Even when hidden within crevices, their bodies release a continuous stream of chemical cues into the surrounding water. Mucus secreted from the skin contains amino acids and other compounds that dissolve into the water and spread outward through subtle currents. To a predator equipped with sensitive olfactory organs, these signals function like a trail leading directly to the source. A resting fish may appear invisible in the darkness, yet its scent betrays its location. Parrotfish confront this challenge with a strategy that seems almost surreal when first observed. As the light fades and the reef enters its nocturnal phase, an individual parrotfish begins preparing for sleep. Rather than seeking the deepest crevice available, the fish settles into a sheltered spot within the reef and begins producing mucus from specialized glands located in its mouth. The fish rotates slowly while releasing this mucus in a continuous stream, spreading it outward until it forms a thin, transparent envelope surrounding its entire body. Within minutes, the fish becomes encased in a nearly spherical cocoon composed entirely of mucus. The membrane floats slightly above the skin, forming a delicate bubble that isolates the fish from direct contact with the surrounding water. Only the faint movement of the fish's gills disturbs the stillness inside the enclosure as it settles into a state of rest. The cocoon remains almost invisible unless illuminated at close range, appearing like a fragile film suspended around the fish's body. The biological function of this structure reveals itself when considering the sensory abilities of nocturnal predators. Moray eels detect prey primarily through chemoreception, using specialized nasal organs to analyze dissolved chemical signals drifting through the water. By surrounding itself with a mucus envelope, the parrotfish effectively traps its own scent inside a contained environment. The cocoon acts as a barrier that slows the diffusion of chemical cues into the surrounding water. Instead of releasing a continuous stream of scent molecules that would guide predators directly to its resting place, the fish isolates those signals within the bubble. The result is a form of chemical stealth. A Moray eel gliding through the reef at night may pass within a short distance of a sleeping parrotfish without detecting its presence. Without the telltale chemical trail that normally reveals resting fish, the predator continues searching elsewhere. The cocoon does not make the fish physically invisible, but it disrupts the sensory channel that the predator depends upon most strongly. Researchers studying parrotfish behavior have proposed that the cocoon may also serve additional protective functions. The mucus barrier could deter small parasites that attempt to settle on the fish's skin during the night. Reef environments contain numerous tiny crustaceans that search for hosts while fish remain stationary. By enveloping itself in a slippery membrane, the parrotfish may reduce the ability of these organisms to attach successfully. Another intriguing possibility involves early detection of predators. The mucus envelope is delicate enough that even a slight disturbance may rupture it. If a nocturnal predator brushes against the cocoon while investigating the area, the sudden break in the membrane may trigger a sensory response in the resting fish. This disturbance could provide the crucial fraction of time needed for the parrotfish to awaken and escape before the predator locates its exact position. The production of the cocoon itself reflects the remarkable adaptability of parrotfish physiology. The mucus is secreted from glands within the mouth and released gradually as the fish rotates its body, spreading the material evenly around its surface. The process requires both precise timing and coordination. Too little mucus would leave gaps in the barrier, while excessive secretion could create a structure too fragile to remain intact throughout the night. The fish appears to regulate the process carefully, producing just enough material to form a stable enclosure that persists until morning. As darkness deepens across the reef, hundreds of parrotfish settle into their chosen resting places and construct similar cocoons. Each bubble glimmers faintly in the moonlit water, suspended like delicate spheres among the coral branches. Within these fragile shelters, the fish remain almost motionless, conserving energy, while the nocturnal phase of the ecosystem unfolds around them. Beyond the resting parrotfish, the reef transforms into a completely different environment. Crustaceans emerge from their hiding places to forage across the coral surface. Octopuses extend their arms through crevices, searching for crabs and shrimp. Moray eels glide slowly between coral towers, their heads weaving from side to side as they sample the water for traces of prey. Every movement carries chemical signals that drift through the water column, forming an invisible web of scent trails. In this chemical landscape, the parrotfish cocoon functions as a remarkable evolutionary innovation. Rather than attempting to overpower predators through speed or defensive weapons, the fish alters the sensory information available to its hunters. By blocking the release of scent molecules, it disrupts the predator's ability to detect its presence in the first place. This strategy represents a subtle but effective adaptation to the nocturnal challenges of reef life. As the night progresses, the reef continues its hidden activity beneath the cover of darkness. Predators pursue prey guided by smell and vibration, while countless small organisms participate in feeding cycles that rarely occur during daylight hours. The parrotfish remain safely enclosed within their mucus bubbles, suspended between coral branches like sleeping figures protected by invisible shields. When dawn finally approaches and the first faint light returns to the water, the reef begins its transformation once again. Nocturnal predators retreat into crevices, while daytime fish prepare to resume their activity. Inside the mucus cocoon, the parrotfish stirs as the surrounding light brightens. With a few quick movements, it breaks through the delicate membrane and swims free, leaving fragments of the cocoon drifting slowly away in the current. Within moments, the fish returns to its familiar routine of grazing across coral surfaces, scraping algae with its powerful beak, and contributing once more to the maintenance of the reef ecosystem. The mucus cocoon, which served as a crucial protective strategy throughout the night, disappears entirely as the reef shifts back into its daytime rhythm. In the intricate world of coral reefs, survival often depends not only on physical strength or speed, but also on the ability to manipulate the flow of information within the environment. The parrotfish cocoon represents one of the most elegant examples of this principle. Through a simple yet effective biological innovation, the fish transforms its vulnerability during sleep into a form of chemical invisibility. Within the reef's nightly drama of predator and prey, this fragile bubble becomes a silent shield that allows one of the ecosystem's most important herbivores to rest undetected until the sun rises again. Where rivers meet the sea, the water begins to lose its clarity. Salt and fresh water mix in swirling layers that carry sediment from distant mountains and nutrients washed from forests and wetlands. The color shifts from the deep blue of open ocean to a cloudy green or brown, and the scent of mangroves, mud, and decaying leaves fills the air above the surface. Beneath that surface, the ecosystem changes just as dramatically. Fish that evolved in salt water mingle with species adapted to freshwater currents, and the boundary between two worlds becomes blurred. Estuaries are often described as nurseries for marine life, but they are also corridors through which certain predators move silently into territories that few ocean hunters ever reach. Along the mouth of a wide tropical river, a large shadow glides beneath the surface. At first it seems out of place among the freshwater fish that dart through submerged branches and driftwood. Yet this predator is perfectly adapted to the strange chemistry of this environment. It is a bull shark, one of the few sharks on Earth capable of moving freely between the saltwater ocean and the freshwater rivers that penetrate deep into continental interiors. For most marine animals, such a journey would be lethal. The chemistry of freshwater disrupts the delicate balance of salts and fluids inside their bodies, causing cells to swell and organs to malfunction. Yet the bull shark crosses this boundary with remarkable ease. Its physiology equipped with a system that allows it to adjust to radically different environments within a matter of minutes. The river stretches inland through dense tropical forest, carrying the faint influence of ocean tides far beyond the coastline. Mudbanks collapse slowly into the current, while roots from riverside trees form underwater tangles where small fish seek shelter. These fish belong to communities that have evolved without constant exposure to large marine predators. In the open ocean, prey species often develop specialized behaviors to evade sharks. Schooling patterns, rapid bursts of speed, and heightened sensitivity to water vibrations all provide a measure of protection. In rivers, however, the evolutionary arms race has unfolded differently. Freshwater predators such as catfish and crocodilians rely on stealth and ambush rather than the swift pursuit tactics common among oceanic hunters. When a bull shark enters this environment, it introduces a style of predation that many river species have never encountered. The bull shark's ability to penetrate these inland waters begins with a physiological challenge known as osmoregulation. Marine fish live in an environment where the surrounding water contains far more dissolved salt than their internal body fluids. To maintain balance, they must prevent excessive water loss while excreting the surplus salts absorbed through their gills. Freshwater fish face the opposite problem. The surrounding water is less salty than their bodies, causing water to flow inward through tissues while valuable salts diffuse outward. Each group has evolved mechanisms suited to its particular environment, but very few animals can switch between these conditions successfully. Bull sharks achieve this flexibility through a remarkable set of internal adjustments centered largely in their kidneys and associated glands. When the shark inhabits salt water, its body maintains high concentrations of urea and other compounds that help balance the osmotic pressure between internal fluids and the surrounding ocean. Specialized cells in the gills and kidneys regulate the movement of salts, preventing dehydration while maintaining stable blood chemistry. As the shark begins moving into fresh water, however, this balance must change rapidly. The surrounding water now threatens to dilute the salts inside the shark's body while flooding tissues with excess fluid. Within minutes of entering fresh water, the bull shark's kidneys alter their processing mode. Instead of conserving water and excreting excess salts, the organs begin producing large volumes of dilute urine that remove incoming water from the bloodstream. At the same time, the shark reduces the concentration of urea circulating through its body. This shift lowers the osmotic pressure of its internal fluids, preventing water from rushing uncontrollably into its cells. The process resembles a biological switch, allowing the shark to adjust its internal chemistry to match the conditions of the river environment. Other organs participate in this transformation as well. The rectal gland, which normally secretes concentrated salt solutions when the shark lives in the ocean, dramatically reduces its activity in fresh water. Specialized ion transport cells within the gills change their behavior, conserving salts that would otherwise diffuse into the surrounding water. Through the coordinated activity of these systems, the bull shark maintains the chemical stability of its body even as it moves between habitats that would destroy most marine predators. The evolutionary advantage of this capability becomes clear as the shark travels deeper into the river. The water grows murkier and slower, carrying floating debris and suspended sediment that limit visibility. Many river fish rely more on lateral line systems and chemical detection than on vision. Yet the bull shark possesses sensory equipment refined in the vast hunting grounds of the ocean. Electroreceptors embedded in its snout detect faint electrical signals produced by the muscles and nerves of nearby animals. These receptors allow the shark to locate prey hidden beneath muddy water or concealed among submerged branches. As the shark patrols the river channel, schools of small fish scatter ahead of it, sensing the disturbance of water displaced by its powerful body. Larger fish react more slowly, unaccustomed to predators that approach with such speed and force. In the open ocean, prey species have evolved to recognize the distinctive silhouette and movement patterns of sharks. In the river environment, those cues may not trigger immediate alarm. The result is a temporary imbalance in the ecological arms race, giving the bull shark an advantage that few freshwater predators can match. Evidence of these inland journeys has been documented across multiple continents. Bull sharks have been recorded hundreds of miles from the sea, navigating rivers that twist through forests, farmland, and even densely populated cities. In some cases, individuals have traveled so far upstream that their presence seems almost impossible, yet the biological mechanisms supporting their migration remain remarkably efficient. The ability to alter kidney function and internal chemistry allows these sharks to move between ecosystems that were once considered completely separate. For the bull shark, these river systems offer several potential benefits. In some regions, freshwater habitats provide nursery grounds where young sharks can grow with reduced competition from larger oceanic predators. The shallow, turbid waters limit visibility, giving juvenile sharks protection while they develop hunting skills. For adults, rivers can also serve as seasonal feeding grounds, rich with fish that gather to spawn or migrate. Within the muddy current of a tropical river, the shark moves with deliberate calm. Its body sways gently as it follows the contours of the channel, occasionally brushing against drifting branches or submerged logs. The water around it carries countless chemical signals released by fish, amphibians, and other aquatic animals. Each faint electrical pulse detected by the shark's electroreceptors reveals another potential target hidden within the murk. A group of freshwater fish hovers near the edge of a submerged sandbank, feeding on small invertebrates stirred up by the current. Their attention remains focused on the sediment below, unaware of the approaching predator. The bull shark glides closer, its broad head sweeping slightly from side to side as it interprets the electrical patterns radiating from the school. With a sudden burst of acceleration, the shark lunges forward. Water explodes outward as the predator slices through the group, seizing a fish before the others can scatter. Moments later, the river returns to its quiet rhythm. Suspended sediment settles slowly through the water column while the surviving fish regroup near the bottom. For them, the attack arrives as a shock, a brief encounter with a predator whose evolutionary roots lie far beyond the river's boundaries. Yet, the bull shark continues its patrol, its physiology fully adapted to the freshwater environment through the remarkable flexibility of its kidneys and associated systems. The ecological implications of this ability extend beyond individual hunting events. By entering rivers and estuaries, bull sharks connect marine and freshwater ecosystems in ways that few other large predators can replicate. Their presence influences the behavior of fish populations, altering feeding patterns and habitat use. Prey species may begin avoiding certain areas of the river channel, redistributing themselves into shallower zones or dense vegetation where the shark's large body cannot maneuver as easily. Over evolutionary timescales, these interactions may shape the development of defensive behaviors among freshwater species. Yet such adaptations take generations to emerge. In many regions where bull sharks appear only occasionally, the local fish populations remain poorly prepared for this type of predator. The result is a fleeting advantage for the shark, a momentary imbalance in the ongoing dialogue between predator and prey. As the river bends through the forest and begins its long journey back toward the sea, the bull shark eventually follows the current downstream. The chemical composition of the water changes gradually as salinity increases, signaling the approach of the ocean. Once again, the shark's internal systems begin shifting. Kidney function adjusts to reduce water loss, urea concentrations rise within the bloodstream, and the rectal gland resumes its role in removing excess salt. Within a short time, the shark's physiology returns to the configuration suited for life in the open ocean. When the shark finally reaches the coastal waters, where river and sea merge, the environment broadens once more into the vast blue domain of marine predators. Yet the memory of its inland journey remains encoded within its biological machinery. Through the remarkable flexibility of its kidneys and the coordinated systems that regulate its internal chemistry, the bull shark remains one of the few apex predators capable of bridging two worlds that most species experience only separately. In the ever-shifting boundary where rivers meet the ocean, this adaptability grants the bull shark a unique ecological role. It is both a marine hunter and a freshwater infiltrator, a creature whose physiology allows it to cross environmental barriers that limit nearly every other shark. Through this genetic and physiological innovation, the bull shark expands its hunting territory far beyond the familiar expanses of the sea, carrying the influence of an ocean predator deep into the winding arteries of the continents themselves. Along the temperate coastlines of southern Australia, the ocean feels different from the bright tropical reefs farther north. The water carries a cooler weight, rich with nutrients that rise from deeper currents and feed vast underwater forests. Here, towering stands of kelp grow from rocky sea floors, stretching upward toward the light like submerged trees. Their broad fronds twist and ripple constantly in the movement of the sea, transforming the water column into a slow-motion canopy of swaying vegetation. Sunlight filters through the floating blades in shifting green patterns, creating a landscape where visibility changes with every passing wave. For animals living within these forests, survival depends not only on strength or speed, but on the ability to vanish into the moving architecture of the kelp itself. In this environment, the water is rarely still. Even on calm days, the surge of distant swells pushes gently through the kelp forest, causing entire stands of seaweed to lean and recover in slow, rhythmic motions. Every blade moves with the same pulse, bending together before rising again, a silent choreography driven by the ocean's breathing currents. Among these shifting fronds swims a creature that appears less like an animal and more like a fragment of the forest itself. Drifting between the kelp blades is the leafy sea dragon, an animal so intricately disguised that it often disappears even under close observation. At first glance, it resembles nothing more than a cluster of torn seaweed floating free in the current. Thin, leaf-like appendages extend from every part of its body, branching outward in delicate shapes that mimic the irregular edges of kelp fronds. The creature's body is slender and elongated, with a narrow snout and small fins that move almost imperceptibly. Yet it is the leafy projections that dominate the animal's silhouette, transforming its outline into a living piece of vegetation. Unlike many animals that rely on camouflage by changing color, the leafy sea dragon embodies disguise in its physical structure. Each leaf-like appendage is not an external attachment or piece of debris caught on the body. They are true extensions of the animal's skin, genetic structures that develop as part of its anatomy. These protrusions contain no muscles for propulsion and play no role in swimming. Their sole purpose is visual deception. By breaking up the sea dragon's outline and mimicking the shape of surrounding seaweed, they render the animal nearly invisible within its habitat. The effectiveness of this disguise becomes clear when observing how predators navigate the kelp forest. Fish that hunt among these underwater plants must constantly distinguish between living prey and the shifting vegetation around them. Movement provides one of the most reliable clues. A fish that darts suddenly through the water stands out against the slower motion of swaying kelp. For this reason, many animals living in kelp forests move cautiously, adjusting their behavior to match the rhythm of the environment. The leafy sea dragon takes this principle to an extraordinary level. Its small fins, located along the neck and near the tail, beat so gently that the animal appears to drift rather than swim. The motion of its body mirrors the slow oscillations of the kelp blades around it. When the current pushes the seaweed to one side, the sea dragon tilts slightly in the same direction. When the fronds recover and rise again, the animal follows that movement with subtle adjustments. From a distance, the sea dragon becomes indistinguishable from the surrounding vegetation. This synchronization with the environment extends beyond simple imitation. The frequency and amplitude of the sea dragon's movements closely match those of the kelp forest itself. Ocean currents passing through the forest create predictable patterns of sway determined by water velocity, blade flexibility, and the density of vegetation. Over evolutionary time, the sea dragon's behavior has adapted to align with these patterns, ensuring that its motion remains consistent with the background environment. To a predator scanning the forest for unusual movement, the sea dragon offers no obvious signal of life. Despite its elaborate disguise, the leafy sea dragon remains an active hunter within this ecosystem. Its diet consists primarily of small crustaceans, including mysid shrimp and tiny amphipods that drift through the kelp forest in loose swarms. These animals rely on their own forms of camouflage and rapid movement to avoid predators. For them, the forest represents both a refuge and a feeding ground, a place where plankton and detritus accumulate among the fronds. The sea dragon approaches its prey with extraordinary patience. Rather than chasing shrimp through the water, it glides slowly through the forest, allowing currents to carry it among the drifting crustaceans. Its long tubular snout functions as a precise feeding tool. When a shrimp wanders within range, the sea dragon expands its throat cavity suddenly, creating a brief suction that draws the prey into its mouth. The movement is so subtle that nearby animals often fail to detect the attack until it has already occurred. Unlike many predatory fish, the sea dragon lacks teeth and powerful jaws. Instead, its feeding strategy depends on stealth and precision. The camouflage that protects it from predators also allows it to approach prey unnoticed. By blending perfectly into the kelp forest, the sea dragon becomes a drifting ambush hunter that captures food with minimal effort. The anatomy supporting this lifestyle reveals further evolutionary specialization. The leafy appendages covering the sea dragon's body are supplied with blood vessels that allow them to maintain their shape and coloration. Their thin surfaces catch light in ways similar to actual seaweed blades, producing subtle reflections that enhance the illusion of vegetation. At the same time, the sea dragon's body coloration ranges from pale green to yellowish brown, closely matching the hues of kelp and other marine plants. Yet camouflage alone does not guarantee survival. The kelp forest hosts numerous predators capable of detecting prey through senses other than sight. Larger fish patrol the outer edges of the forest, while seals and seabirds occasionally dive into the vegetation searching for food. In such a dynamic environment, the sea dragon's passive defense must remain effective under varying conditions of light and water movement. One factor contributing to its success is the sheer complexity of the kelp forest itself. The dense arrangement of fronds creates visual noise that overwhelms the sensory systems of many predators. Shapes overlap, shadows shift constantly, and reflections from the water surface distort the outlines of objects below. Within this chaotic visual field, the sea dragon's leaf-like body becomes just another fragment of the forest. Interestingly, the appendages that give the sea dragon its remarkable disguise do not contribute to swimming. Instead, propulsion comes from two nearly transparent fins that vibrate rapidly but with extremely small amplitude. One fin runs along the back of the animal, while another sits near the neck. These fins move so quickly and subtly that they are almost invisible in the water. The result is a mode of locomotion that avoids sudden displacement of water, further reducing the chance that nearby animals will detect the sea dragon's presence. The kelp forest itself benefits from the presence of animals like the leafy sea dragon. By feeding on small crustaceans, the sea dragon participates in a complex food web that regulates the abundance of plankton feeders within the forest. This balance influences how nutrients circulate through the ecosystem and how energy moves between trophic levels. Although the sea dragon appears delicate and passive, it occupies a meaningful role within the broader ecological network. Reproduction introduces another fascinating aspect of the species' biology. Unlike most fish, the female sea dragon does not carry eggs internally. Instead, after courtship, she transfers a cluster of bright pink eggs onto the underside of the male's tail. Specialized tissues on the tail hold the eggs securely, supplying oxygen and protection as the embryos develop. For several weeks, the male carries this living cargo while continuing to move through the kelp forest with careful precision. During this time, the camouflage becomes even more important. The eggs add weight and color to the male's body, potentially increasing the risk of detection by predators. Yet the leafy appendages continue to mask his outline effectively. As currents sway the kelp around him, the egg-bearing male drifts through the forest like a floating cluster of seaweed carrying hidden life within it. Eventually the eggs hatch, releasing tiny sea dragons into the water column. These juveniles must begin life immediately within the same complex environment that shaped their parents' evolution. Though much smaller, and lacking the full development of leafy appendages, they already display behaviors that mirror the motion of surrounding vegetation. Even at this early stage, the strategy of blending with the forest begins guiding their survival. Over evolutionary timescales, the leafy sea dragon has refined its disguise into one of the most extraordinary examples of passive defense in the natural world. Rather than fleeing from predators or confronting them with venom or armor, the animal relies entirely on invisibility. Its body has become an extension of the kelp forest itself, shaped by genetics to replicate the form and movement of the plants that dominate its habitat. As currents continue to sweep through the underwater forest, the sea dragon drifts slowly among the kelp blades. From a distance, it is impossible to distinguish where plant ends and animal begins. The leaves ripple gently with the surge, sunlight flickers across their surfaces, and the forest breathes with the steady rhythm of the sea. Somewhere within that motion, a predator may pass within inches of the sea dragon, scanning the water for signs of life. Yet the predator sees only seaweed. In the quiet complexity of the kelp forest, the leafy sea dragon exists as a ghost woven into the landscape. Every protrusion, every motion, every color pattern works in harmony with the surrounding vegetation. Through this extraordinary botanical illusion, the animal transforms vulnerability into invisibility, becoming not merely a resident of the kelp forest, but an indistinguishable part of the living underwater forest itself. The continental shelf slopes gently away from the coastline, descending into a dim world where sunlight fades into blue shadow long before it reaches the seabed. Here the ocean floor is scattered with rock outcrops, narrow crevices, and fields of coarse sand sculpted by slow-moving currents. Seaweed drifts above the rocks like underwater banners, while brittle stars and sea urchins creep across the sediment in patient, almost mechanical motions. This environment does not reveal its secrets easily. Most animals living here emerge only briefly, retreating into cracks and tunnels whenever danger passes overhead. Among these hidden inhabitants lives a creature that has survived in this manner for millions of years, navigating the cold, shadowed seafloor with a strategy that appears almost impossible from a biological perspective. From beneath a slab of rock protrude two long antennae that sweep slowly through the water like sensing rods. Their motion is deliberate, probing the surrounding current for chemical traces and vibrations that reveal the presence of food or predators. Gradually, a heavy armored body follows, inching forward on jointed legs until the full shape of a lobster emerges into view. Its exoskeleton is thick and segmented, colored in mottled tones of deep brown and muted blue that match the rocky substrate beneath it. The lobster pauses briefly, adjusting its antennae once more before continuing its slow patrol along the seafloor. At first glance, the lobster appears to represent a familiar strategy among marine animals. Hard armor protects its soft tissues, and powerful claws allow it to defend itself or manipulate prey. Yet beneath that sturdy shell lies one of the most unusual biological phenomena in the animal kingdom. Unlike most creatures, lobsters do not age in the traditional sense. Their cells possess a mechanism that allows them to maintain genetic integrity far longer than the cells of most animals can endure. This mechanism revolves around an enzyme known as telomerase, a molecular repair system that preserves critical structures within the DNA of the lobster's cells. To understand why this matters, it helps to consider the way most organisms age. Inside every cell of a living animal lies a set of chromosomes carrying genetic instructions encoded in DNA. At the ends of these chromosomes exist structures called telomeres, sequences of DNA that function like protective caps. Each time a cell divides to create new cells, a small portion of these telomeres is lost. Over time, the telomeres become progressively shorter until the cell can no longer divide safely. This gradual erosion contributes to the aging process, limiting the ability of tissues to repair themselves and maintain healthy function. In many animals, the enzyme telomerase appears only during early development and then declines dramatically as the organism matures. Without this enzyme, the telomeres continue shrinking with each cell division. Eventually, the loss becomes severe enough that cells enter a state known as senescence, where they remain alive but can no longer divide or perform their functions effectively. The accumulation of senescent cells contributes to the weakening of tissues and organs that characterizes aging. Lobsters operate under a different biological rule. Their bodies maintain unusually high levels of telomerase activity throughout life. The enzyme continuously rebuilds the telomeres at the ends of chromosomes, repairing the damage that would otherwise accumulate through repeated cell division. As a result, the lobster's cells retain the capacity to divide and regenerate far longer than those of most animals. In theory, this means that the lobster does not experience the same gradual cellular aging that affects mammals, birds, or many other marine species. This phenomenon has led scientists to describe lobsters as biologically immortal, though the phrase requires careful interpretation. Immortality in this context does not mean that a lobster lives forever. Rather, it means that the animal does not possess a built-in biological clock that inevitably leads to senescence in the way it does for most organisms. A lobster can continue growing and repairing its tissues for decades, sometimes more than half a century under favorable conditions. Growth in lobsters occurs through a process known as molting, an event that defines nearly every stage of their life cycle. Because the exoskeleton cannot expand once it has hardened, the lobster must periodically shed its entire shell to grow larger. Before molting begins, the lobster absorbs minerals from the existing shell, storing them internally, while a new soft exoskeleton forms beneath the old one. At the critical moment, the old shell splits along a seam behind the head, and the lobster slowly pulls itself free. For a brief period following the molt, the lobster's body remains soft and vulnerable. The new exoskeleton gradually hardens as minerals are redeposited, restoring the armor that protects the animal from predators. During this vulnerable phase, the lobster often hides within crevices or burrows, minimizing movement until its shell regains strength. As the new shell solidifies, the lobster emerges slightly larger than before, continuing a cycle of growth that may repeat many times throughout its life. This molting process explains both the remarkable longevity of lobsters and the ultimate limitation that defines their lives. Because telomerase preserves the ability of cells to divide and repair tissues, the lobster's body continues growing indefinitely. Each successful molt allows the animal to increase in size, sometimes reaching impressive proportions after decades on the seafloor. However, molting itself requires enormous energy. The lobster must generate new tissue, build a new shell, and survive the vulnerable period before that shell hardens. As the lobster grows larger, the energy demands of molting increase dramatically. Larger bodies require thicker shells, more muscle, and greater metabolic investment to complete the process. Eventually, the lobster reaches a size at which the energy required to molt exceeds the energy it can realistically obtain from its environment. At this point, the animal faces a biological paradox. Its cells remain capable of regeneration thanks to telomerase, yet its body can no longer complete the molting process necessary for continued growth. When that threshold is reached, the lobster may attempt a final molt that fails to complete successfully. Without enough energy to finish building and hardening the new exoskeleton, the animal becomes trapped in a vulnerable state. Predators, disease, or simple exhaustion often end the lobster's life at this stage. In a sense, the lobster does not die because it grows old in the traditional biological sense. Instead, it dies because its remarkable growth eventually exceeds the energetic limits of its environment. On the seafloor, where this lobster patrols, such outcomes are rarely visible. Most lobsters live within networks of rocky shelters, where they avoid many predators through caution and concealment. Their antennae constantly sample the water for chemical cues released by nearby animals. When the scent of carrion drifts through the current, the lobster may follow the trail to a dead fish or other organic debris, feeding with deliberate movements of its claws. Although lobsters are often associated with scavenging, they also function as opportunistic predators within the benthic ecosystem. Small mollusks, worms, and crustaceans become targets when the lobster encounters them. The crushing power of its claws allows it to break open shells or seize prey hiding among rocks. Through these feeding habits, the lobster participates in the recycling of nutrients on the seafloor, converting dead or slow-moving organisms into energy that supports its own long lifespan. The lobster's sensory abilities complement its armored body and unusual longevity. Its antennae contain receptors capable of detecting extremely faint chemical traces in the water. These signals guide the animal toward food sources or warn it of approaching predators. Tiny hairs along the antennae also sense subtle water movements, allowing the lobster to detect disturbances long before a threat comes into view. As evening approaches and the light above the seafloor dims further, the lobster continues its steady exploration among the rocks. Nearby, fish glide through the water column, while sea stars creep slowly across the sand in search of clams buried beneath the sediment. The ecosystem functions as a network of interactions where each species influences the others through feeding, competition, and habitat modification. Within this network, the lobster occupies a unique position, not only because of its armored body, but because of its extraordinary cellular biology. The enzyme telomerase quietly performs its work inside millions of cells, repairing the ends of chromosomes and preserving the genetic integrity required for continued growth. While other animals age through the gradual breakdown of cellular machinery, the lobster's tissues maintain their capacity for renewal far longer than most. Yet the environment ultimately imposes its own limits. Energy availability, predation, disease, and the mechanical challenges of molting all shape the life trajectory of even the most biologically resilient organisms. The lobster's story illustrates that longevity does not necessarily guarantee immortality. Instead, it reveals how evolutionary adaptations can push the boundaries of life's durability while still remaining bound by the physical realities of growth and energy. As currents sweep gently across the rocky seabed, the lobster disappears once more into the shadows of its shelter. The antennae withdraw, and only the faint outline of its armored carapace remains visible in the dim light filtering from above. Within that shell lives a creature whose cells continue repairing themselves year after year, quietly extending a lifespan that may surpass many of the animals sharing its habitat. In the cold stillness of the ocean floor, the lobster represents a remarkable biological anomaly. It does not age in the way most animals do. Instead, it grows, molts, and renews itself repeatedly until the demands of that endless growth finally exceed what the environment can sustain. In this way, the lobster carries within its DNA what might be called an immortality glitch, a molecular quirk that allows life to persist far beyond ordinary limits until the simple physics of size and energy bring the long journey to an end. The tide has retreated from the rocky coastline, leaving behind a shallow mosaic of pools scattered across the exposed shore. These tide pools are temporary worlds, isolated pockets of seawater trapped between jagged rocks and ridges of coral skeletons. Sunlight penetrates them easily, revealing miniature ecosystems where an extraordinary number of organisms coexist within a space no larger than a small room. Tiny fish weave between patches of algae, while crabs patrol the edges with careful steps. Sea anemones expand their tentacles toward the light, waiting for drifting particles of food carried by gentle currents that seep in from the open ocean beyond the rocks. Within this confined but vibrant habitat, survival often depends on resilience. Waves crash overhead with unpredictable force, temperatures fluctuate throughout the day, and predators arrive whenever the tide rises high enough to reconnect these isolated pools with the wider sea. Spread across the rock surface at the bottom of one of these pools lies a starfish, its 5 arms extended outward like the spokes of a wheel. At first glance it appears almost motionless, a quiet presence against the textured stone. Its body is rough to the touch, covered with tiny calcified plates and small protruding spines that form part of its external skeleton. Beneath this armor lies one of the most unusual anatomical designs found in the animal kingdom. Unlike most creatures whose bodies rely on centralized organs and a dominant brain controlling the entire system, the starfish operates with a decentralized architecture. Each arm functions as a semi-independent unit containing portions of the digestive system, reproductive tissues, and nerve networks necessary for survival. The tide pool around the starfish is filled with life. Small mollusks cling tightly to the rocks, grazing on microscopic algae that coat the surfaces. Barnacles filter water for plankton, while limpets scrape the stone with rasping mouthparts. These animals may appear stationary, but they remain alert to the presence of predators capable of prying them loose from their secure footholds. Among the most persistent of these predators is the starfish itself, which moves slowly but with remarkable determination. The starfish begins to crawl across the rock, using hundreds of tiny tube feet located along grooves on the underside of its arms. Each tube foot operates through a hydraulic system powered by seawater drawn into the body through a small structure called the madreporate. By adjusting pressure within this internal water vascular system, the starfish extends and retracts the tube feet in coordinated waves that pull the body forward. The movement is slow, almost imperceptible from moment to moment. Yet over time, the animal steadily advances across the rock surface. As the starfish approaches a cluster of muscles attached to the stone, its arms begin to curve gently around the group. Muscles defend themselves by clamping their shells tightly together, relying on strong adductor muscles that hold the valves closed against most predators. The starfish counters this defense with patience rather than brute force. Using the adhesive tips of its tube feet, it applies steady pressure along the edges of the shells, pulling in opposite directions with multiple arms. The effort may last hours, but eventually the muscles inside the muscle fatigue and a narrow gap opens between the shells. Through this tiny opening, the starfish deploys one of its most remarkable feeding adaptations. Instead of pulling the prey into its mouth, the starfish pushes its stomach outward through the opening and into the shell. The stomach envelops the soft tissues of the muscle, releasing digestive enzymes that begin breaking down the prey externally. Once digestion is complete, the liquefied nutrients are absorbed back into the starfish's body as the stomach retracts. This unusual feeding strategy allows the starfish to consume prey that would otherwise remain inaccessible within sealed shells. Yet the most extraordinary aspect of the starfish lies not in its feeding behavior, but in the organization of its body itself. At the center of the animal lies a disc where the arms converge. Within this disc are organs associated with digestion and reproduction, but the nervous system is distributed in a ring that extends into each arm. Rather than relying on a single centralized brain, the starfish processes sensory information through this network of interconnected nerves. Each arm contains sensory cells capable of detecting light, touch, and chemical cues from the surrounding water. This decentralized design offers remarkable advantages when the animal faces injury or predation. In the tide pool environment, many predators target starfish, including certain fish, birds, and even other invertebrates. When threatened, some starfish species possess the ability to detach one of their arms deliberately, sacrificing the limb to distract the attacker while the rest of the body escapes. The detached arm may continue moving briefly, confusing the predator long enough for the main body to crawl away to safety. Unlike most animals, however, the story does not end with the loss of a limb. The starfish possesses an extraordinary capacity for regeneration. Over time, the remaining body can grow a new arm to replace the one that was lost. Even more astonishing is what can happen to the detached limb itself. Because each arm contains portions of the digestive system and nervous network, it may remain viable long enough to begin regenerating the rest of the body. Given sufficient resources and time, the severed arm can develop a new central disc and additional arms, effectively producing a second starfish genetically identical to the original. This process represents a form of biological cloning, achieved not through reproduction, but through regeneration. The genetic information contained within the tissues of the arm guides the growth of missing structures until a complete organism emerges. For species that inhabit environments where injuries are common, this ability offers a powerful survival advantage. Even catastrophic damage does not necessarily result in death if enough tissue remains capable of initiating the regeneration process. Inside the starfish's body, specialized cells orchestrate this rebuilding effort. When an arm is lost, cells near the wound begin dividing rapidly, forming a mass of undifferentiated tissue known as a regeneration blastema. These cells gradually specialize into different types required for rebuilding structures such as muscles, skeletal plates, nerves, and tube feet. Over weeks or months, the missing limb grows outward from the central disc until it matches the original size and function of the other arms. The energy required for regeneration is substantial, and the process depends heavily on environmental conditions. Starfish living in nutrient-rich habitats with abundant prey can regenerate limbs more quickly than those in environments where food is scarce. Temperature also influences the rate of cellular activity. Warmer waters often accelerate tissue growth, while colder conditions slow the regeneration process dramatically. Despite its apparent simplicity, the starfish's decentralized anatomy creates a form of biological redundancy rarely seen among animals. Because vital systems extend into multiple arms rather than being confined to a single location, damage to one region does not necessarily compromise the entire organism. This distributed design resembles certain modern technological systems in which multiple processors share tasks across a network. If one component fails, others continue functioning until repairs are made. Within the tide pool, the starfish continues its slow exploration of the rocky surface. Nearby, a crab scuttles between algae patches while a small fish darts through the water column above. Each organism participates in the delicate balance that defines life in this narrow strip between ocean and land. Predation, competition, and environmental stress shape the behavior of every species living here. The starfish's regenerative ability also influences its interactions with predators. Some animals may consume individual arms without eliminating the entire starfish population. In such cases, the prey species effectively recovers from what would be a fatal injury for most animals. Over evolutionary time, this resilience may alter the dynamics between predator and prey, allowing starfish populations to persist despite frequent attacks. At the microscopic level, the regenerative process reveals fascinating insights into cellular plasticity. Cells within the starfish possess the ability to revert to less specialized states before transforming into new tissues. This capacity has drawn interest from scientists studying regenerative medicine, as it demonstrates how complex body structures can reassemble themselves from relatively simple cellular beginnings. The tide begins to rise again as waves from the open ocean push fresh water into the rocky pools. The boundary between isolated ecosystems dissolves, reconnecting the tide pools with the larger marine environment beyond the rocks. Fish that had remained outside the pools during low tide now swim freely among the rocks, exploring areas that were previously inaccessible. The starfish remains anchored to the rock, its tube feet gripping firmly as currents strengthen. For an animal whose life unfolds at such a slow pace, these environmental changes are simply part of the rhythm of existence. Whether feeding on mussels, regenerating lost limbs, or enduring the shifting conditions of the intertidal zone, the starfish embodies a strategy of survival built on resilience rather than speed. Within its seemingly simple form lies a biological system unlike that of most animals on Earth. Vital functions are distributed across multiple arms rather than centralized in a single organ. Injury does not necessarily lead to death, but instead may initiate the creation of an entirely new organism. In this way, the starfish operates like a living network of cooperating parts, each capable of contributing to the survival and regeneration of the whole. As waves continue to wash over the rocks, and sunlight flickers across the surface of the tide pool, the starfish resumes its slow journey across the stone. Beneath the rough surface of its body, countless cells remain ready to repair, rebuild, and regenerate whenever necessary. In the ever-changing environment of the shoreline, this decentralized design transforms vulnerability into resilience, allowing the starfish to persist where many other creatures might fail. The tide has begun to rise over a tropical reef, flat, where broken coral fragments and scattered stones form a landscape that appears almost barren from above. The surface of the seabed looks like a mosaic of rubble. Pale limestone pieces, weathered coral skeletons, patches of coarse sand, and clusters of algae clinging stubbornly to the substrate. Sunlight pours through the shallow water in shimmering ribbons, bending across the uneven terrain as waves pass overhead. Small fish move cautiously between the rocks, pausing frequently to inspect crevices where tiny crustaceans hide. The reef may appear calm, even lifeless in certain patches, yet every stone and coral fragment contributes to a living system where camouflage has become one of the most powerful survival tools available. Among the scattered rocks lies an object that seems no different from the surrounding rubble. Its shape is irregular, its surface textured with bumps and ridges resembling encrusted coral, and its coloration blends perfectly with the muted tones of the seabed. Algae cling to portions of its surface, while fine sediments collect in shallow grooves across its back. To most animals swimming overhead, It is simply another stone resting quietly in the shallow water. Yet this motionless object is not part of the reef's geology. It is a stonefish, an animal whose evolutionary strategy has refined the art of invisibility to a level that approaches perfection. The stonefish occupies a position within the reef ecosystem that depends entirely on deception. Unlike predators that rely on speed, pursuit, or elaborate hunting strategies, the stonefish adopts a radically different approach. It does not chase prey or actively search for food across the reef. Instead, it waits. The fish's body lies pressed against the seabed with its fins spread slightly to anchor it among the rubble. The outline of its body merges seamlessly with surrounding rocks, and its coloration adjusts subtly to match the patterns of algae and sediment nearby. Even experienced divers often fail to notice a stonefish until it moves, which it rarely does. The secret to this extraordinary camouflage lies in the fish's anatomy. Its skin contains specialized pigment cells that allow it to shift shades gradually, blending into the colors of its environment. Irregular skin flaps and wart-like protrusions break up the outline of the body, preventing predators or prey from recognizing the shape of a fish. The head is flattened and broad, with eyes positioned high on the skull, so they remain visible even when most of the body is buried partially in sand. This arrangement allows the stonefish to observe its surroundings while maintaining its illusion as part of the reef. From the perspective of a small fish drifting through the reef flat, the environment appears filled with stationary objects. Coral fragments, shells and rocks lie scattered across the seabed, forming obstacles that fish learn to navigate instinctively. The stonefish exploits this expectation by becoming indistinguishable from those objects. It remains perfectly still for long periods, allowing algae and sediment to accumulate on its body, reinforcing the illusion that it is simply another piece of reef debris. While the stonefish appears passive, its stillness hides an extremely efficient predatory system. The fish's mouth is large and capable of opening with explosive speed. When a small fish or shrimp drifts within range, the stonefish creates a sudden vacuum by expanding its mouth cavity. Water rushes inward, carrying the unsuspecting prey directly into the predator's throat before it has time to react. The entire event lasts less than a fraction of a second. In many cases, nearby animals continue swimming normally, unaware that a predator lies hidden among the rocks. This ambush strategy works precisely because the stonefish avoids detection. In open water environments, prey species often rely on visual recognition of predators to trigger escape responses. On the reef floor, however, the sheer abundance of rocks and debris makes it impossible for small animals to inspect every object carefully. By blending perfectly into this cluttered environment, the stonefish turns the reef itself into a camouflage system that shields its presence. Yet the stonefish's evolutionary design includes another layer of defense far more dramatic than its camouflage. Along its back runs a row of 13 rigid dorsal spines, each connected to venom glands capable of delivering one of the most potent neurotoxins known among fish. These spines remain concealed beneath loose skin and algae-covered tissue until pressure is applied from above. When a predator or unwary animal presses down on the fish's back, the spines pivot upward automatically, penetrating the skin of the intruder and injecting venom through grooves that channel the toxin directly into the wound. The venom of the stonefish functions as a powerful neurotoxin that disrupts nerve signaling and muscle control. Within moments of injection, the toxin interferes with the normal electrical activity required for muscles to contract and relax. In severe cases, the venom can affect the muscles responsible for breathing and cardiac function. For predators that attempt to bite or swallow the stonefish, this defense can prove rapidly debilitating, ensuring that the predator abandons the attack or suffers serious consequences. The ecological significance of this venom extends beyond simple defense. Because the stonefish relies on camouflage rather than mobility, it cannot easily escape predators that detect it. The venom therefore compensates for the fish's lack of speed by creating a powerful deterrent. Predators that experience the effects of the toxin are unlikely to repeat the mistake, reinforcing the survival value of the stonefish's passive strategy. Interestingly, the venom system operates only when physical pressure triggers the spines. The stonefish does not actively strike with its venom in the way that snakes or certain jellyfish deploy their toxins. Instead, The spines act as a reactive mechanism that activates when the fish is stepped on or attacked. This design aligns perfectly with the fish's camouflage strategy. Because the stonefish remains motionless and nearly invisible, most encounters occur accidentally when another animal attempts to rest or move across the seabed. The reef environment surrounding the stonefish is filled with species that must constantly interpret visual signals to survive. Brightly colored fish advertise their toxicity through warning patterns, while others adopt cryptic coloration that blends into coral or sand. The stonefish represents an extreme endpoint of this evolutionary spectrum. Its appearance mimics not merely the color of the reef, but its physical texture and irregular geometry. The animal becomes a perfect imitation of the rocks themselves. Such precise camouflage requires careful alignment between the fish's body structure and its habitat. Stonefish are most commonly found in shallow tropical waters, where coral rubble and algae create complex surfaces. In environments lacking such features, the fish would stand out more clearly, reducing the effectiveness of its disguise. By inhabiting areas rich in visual noise, the stonefish maximizes the probability that predators and prey alike will overlook its presence. As the tide continues to rise, currents begin to move gently across the reef flat, carrying plankton and small organisms through the shallow water. Schools of juvenile fish gather near patches of coral where food particles accumulate. These fish must navigate the terrain carefully, avoiding sudden movements that might attract the attention of larger predators patrolling nearby. Among the rocks, the stonefish remains perfectly still. Only its eyes move slightly, tracking the subtle motion of passing fish. A small goby approaches, pausing momentarily to inspect the algae growing along what it assumes is a piece of coral rubble. The goby's body tilts downward as it searches for microscopic invertebrates hidden among the algae. For an instant, the fish drifts within range of the stonefish's mouth. The predator's jaws open with explosive speed. Water rushes inward, pulling the goby directly into the cavernous mouth before the prey can react. The mouth closes again almost immediately, and the stonefish resumes its still posture among the rocks. Within seconds, the reef returns to its quiet rhythm as though nothing unusual has occurred. For the stonefish, such brief moments of action punctuate long periods of waiting. The fish may remain in the same location for hours or even days, relying on its camouflage and venomous defense to protect it from threats. In this way, the stonefish represents a strategy that pushes passive defense to its absolute limit. Rather than engaging actively with predators or prey, it transforms itself into a nearly invisible part of the environment. This evolutionary approach highlights an important principle within natural ecosystems. Survival does not always depend on speed, strength, or elaborate hunting tactics. Sometimes the most effective strategy is simply to disappear into the background so completely that the surrounding world fails to recognize the danger present. The reef flat gradually deepens as the rising tide reconnects the shallow habitat with the open ocean beyond. Larger fish begin to enter the area, exploring the rubble fields in search of food. Among them swim predators that rely on quick bursts of speed to capture prey in the open water. Yet even these experienced hunters pass within inches of the stonefish without noticing the lethal presence beneath them. To these animals, the stonefish is nothing more than a rock. Only when pressure falls directly upon its back do the hidden spines rise and the venom reveal itself. In those rare moments, the illusion breaks, exposing the extraordinary defense concealed beneath the fish's motionless exterior. But for most of the time, the stonefish remains exactly what evolution designed it to be, an invisible neurotoxin waiting quietly among the stones of the reef, a perfect example of how stillness and deception can transform an ordinary piece of seabed into one of the ocean's most formidable traps. Along the outer edges of the North Pacific coastline, the ocean rarely rests. Long swells roll steadily toward rugged shores where cold currents rising from deep water carry immense quantities of nutrients to the surface. These nutrients fuel dense underwater forests of giant kelp that stretch from the sea floor toward the light like submerged redwood trees. Each blade sways slowly in the constant movement of the sea, forming towering columns of vegetation that transform the water into a layered ecosystem filled with hiding places, feeding grounds, and complex ecological interactions. Within this forest, lives one of the most energetic mammals in the marine world, an animal whose survival depends on maintaining a metabolic engine that runs at extraordinary speed. Floating at the surface among drifting kelp fronds lies a sea otter. Its body appears relaxed as it drifts on its back, forepaws resting loosely on its chest, while the thick fur along its sides traps a shimmering layer of air bubbles. Yet beneath this tranquil posture lies an intense physiological demand. The water surrounding the otter rarely warms beyond temperatures that would quickly sap heat from most mammals. Unlike seals, whales, and other marine mammals that rely on thick layers of insulating blubber, the sea otter possesses almost no body fat. Instead, it survives in these frigid waters through two extraordinary adaptations: the densest fur in the animal kingdom and a metabolism that operates at a pace far faster than that of most mammals of similar size. Each square centimeter of the otter's coat contains hundreds of thousands of hairs, arranged in a dense structure that traps air against the skin. This air layer forms the primary insulation barrier separating the animal's warm body from the surrounding cold water. To maintain that barrier, the otter spends a significant portion of its day grooming. It rolls and twists through the kelp canopy while combing its fur with its paws and teeth, ensuring that the trapped air remains evenly distributed across the coat. Without this constant maintenance, water would penetrate the fur and the animal would lose heat rapidly. Even with such insulation, however, the cold ocean extracts energy continuously from the otter's body. To compensate, the otter's metabolism operates at roughly 3 times the rate expected for a mammal of its size. Its cells burn energy at an extraordinary pace, generating the heat required to maintain a stable internal temperature despite the icy environment. This metabolic furnace comes with a significant cost. The otter must consume an enormous amount of food every day simply to fuel its internal heat production. An adult sea otter may eat approximately 1/4 of its body weight within a single day. For an animal weighing many kilograms, this translates into a constant cycle of foraging that leaves little time for rest. While other marine mammals may spend long periods traveling between feeding events, the sea otter must remain active almost continuously, diving repeatedly into the kelp forest below in search of prey that can sustain its high energy demands. The kelp forest itself provides an ideal hunting ground for such a predator. Beneath the surface, the long fronds anchor to rocky reefs where an abundance of invertebrates thrive among the tangled holdfasts. Sea urchins crawl slowly across the rocks, grazing on algae that coat the surfaces of the reef. Abalone cling tightly to the stone, while crabs patrol the spaces between kelp stems. Clams bury themselves in sediment pockets, while small fish weave through the vertical forest of kelp blades. Each of these species represents potential food for the sea otter, and the otter's survival depends on its ability to harvest them efficiently. When the otter dives, it folds its body gracefully and slips beneath the surface with a powerful kick of its hind feet. Its streamlined form allows it to navigate easily through the dense kelp canopy, using its whiskers to detect subtle water movements that reveal the presence of prey hidden among rocks or buried in sediment. The whiskers are highly sensitive tactile sensors capable of detecting minute vibrations, enabling the otter to locate animals even in the dim underwater light filtered through the kelp. The otter descends toward the seafloor where sea urchins cluster along rocky surfaces. These spiny invertebrates possess a defense that deters many predators, yet the sea otter approaches them with practiced efficiency. Using its dexterous forepaws, it plucks an urchin from the rock and tucks the prey against its chest. Sometimes the otter gathers several urchins at once, storing them temporarily in loose folds of skin beneath its forearms before returning to the surface. Back at the surface, the otter floats on its back and begins the process of opening the spiny shell. In many cases, the otter retrieves a small rock from the seafloor during the dive and carries it upward along with its prey. Resting the stone on its chest like an improvised anvil, the otter strikes the urchin repeatedly against the rock until the shell cracks open. This behavior represents one of the most widely recognized examples of tool use among marine mammals. Once the shell breaks, the otter consumes the nutrient-rich tissue inside, quickly replenishing the energy lost during its dive. This feeding routine repeats dozens of times throughout the day. The otter dives, collects prey, returns to the surface and consumes its catch before descending again. Each cycle provides only a small portion of the energy required to sustain the animal's rapid metabolism. Unlike predators that consume large prey in occasional meals, the sea otter survives through continuous foraging that fills nearly every waking hour. The intensity of this feeding behavior has profound consequences for the kelp forest ecosystem. Sea urchins, one of the otter's preferred prey items, possess an extraordinary appetite for kelp. When urchin populations grow unchecked, they can graze entire kelp forests down to bare rock, creating barren underwater landscapes known as urchin barrens. In regions where sea otters are abundant, however, the constant predation on urchins keeps their numbers in check. This allows kelp forests to flourish, supporting a vast array of species that depend on the vegetation for food and shelter. The sea otter therefore functions as what ecologists describe as a keystone species. Its feeding habits influence the structure of the entire ecosystem. By controlling the population of herbivorous urchins, the otter indirectly protects the kelp forest itself. The towering kelp provides habitat for fish, invertebrates, and marine mammals, creating one of the most productive environments in temperate oceans. Yet maintaining this ecological balance requires relentless effort from the otters themselves. Their high metabolic rate leaves no margin for prolonged fasting. Storms that disrupt foraging or declines in prey availability can quickly place an otter in jeopardy. The animal's body contains little stored fat, meaning that even short periods without sufficient food can lead to rapid weight loss and weakening. Despite these challenges, sea otters have evolved behaviors that help them navigate the energetic demands of their lifestyle. They often anchor themselves within the kelp forest while resting, wrapping strands of kelp around their bodies to prevent drifting away with the current. This habit allows them to sleep safely while floating at the surface, conserving energy between feeding bouts. Mothers with young pups use similar techniques, cradling the pup on their chest while securing themselves to kelp, so that both remain safely within the forest canopy. Reproduction and parental care add another layer of complexity to the otter's energy budget. Female sea otters give birth to a single pup that depends entirely on its mother for nourishment and protection during the early stages of life. The mother must maintain her own energy balance while producing milk rich in fats and nutrients that support the pup's rapid growth. During this period, her already intense foraging efforts increase even further as she gathers enough food to sustain both herself and her offspring. The pup spends much of its time floating at the surface, while the mother dives repeatedly beneath the kelp canopy to gather prey. Because the pup's fur initially contains even more air than that of adults, it floats almost like a buoy, unable to dive until its coat changes during development. The mother often leaves the pup resting on the surface while she hunts below, returning frequently to groom and feed it. As the pup grows, it begins to imitate the mother's behavior, learning to dive and manipulate prey, using its small but increasingly dexterous paws. Over time, the young otter acquires the skills necessary to survive within the demanding environment of the kelp forest, where energy balance determines success or failure on a daily basis. The sea otter's life therefore unfolds as a constant negotiation with the laws of thermodynamics. Cold water continuously drains heat from the body, while metabolism converts food into the warmth needed to maintain life. Every dive, every meal, and every grooming session contributes to the delicate balance that allows the animal to survive in an environment that would overwhelm most mammals. As evening approaches and the light above the kelp forest softens, the sea otter continues its tireless foraging. It dives again into the green shadows below, weaving through columns of kelp, while scanning the sea floor for movement among the rocks. Each successful hunt fuels the metabolic engine that powers its survival. In the quiet complexity of the kelp forest, the sea otter stands as one of nature's most energy-expensive predators. Its body burns fuel at a rate that demands constant effort, transforming every day into a relentless cycle of hunting, feeding, and grooming. Yet through this extraordinary metabolic strategy, the otter not only survives, but shapes the entire ecosystem around it. The kelp forest sways gently in the current above, a living testament to the influence of a small mammal whose internal furnace drives one of the most dynamic ecological relationships in the ocean. Sunlight filters through the upper layers of a tropical coral reef, scattering across the labyrinth of limestone towers and branching corals that stretch across the seabed, like the ruins of a submerged city. Water movement is gentle but constant, carrying microscopic plankton through the reef channels, while fish drift in loose formations between coral heads. Each crevice houses its own community of invertebrates, algae, and small predators that weave together into one of the most biologically complex ecosystems on the planet. In this dense network of life, survival often depends not only on strength or camouflage, but on the ability to adapt quickly when the social structure of a population changes. Among the many species that inhabit the reef, angelfish demonstrate one of the most striking biological responses to such change. A group of angelfish moves slowly along the face of a coral outcrop, their bodies flashing with iridescent blues and yellows as they turn in the shifting sunlight. Their movement appears calm and deliberate. Each fish glides through the water with controlled motions of its fins, stopping occasionally to inspect sponges and algae growing on the reef surface. Although their feeding seems casual, the group maintains a clear internal hierarchy. One individual, larger and more vividly colored than the rest, occupies the central position in the group's movement. This fish is the dominant male, and its presence shapes the behavior of the entire social structure around it. Angelfish communities operate within small territories where a single male typically presides over several females. The group travels and feeds together, but subtle signals maintain the social order. The male patrols the boundaries of the territory, occasionally displaying his size and coloration to remind the others of his status. The females follow loosely behind, feeding and exploring, while remaining within the protective reach of the dominant fish. To an outside observer, the arrangement appears stable, almost routine. Yet beneath the surface lies a biological contingency plan that allows the group to survive even if its leader suddenly disappears. Predators patrol the reef constantly. Larger fish cruise along the reef slopes searching for opportunities to strike, while Moray eels peer from crevices waiting for unsuspecting prey to wander too close. One sudden attack from such a predator can disrupt the delicate social balance of the angelfish group. If the dominant male falls victim to a predator or disappears for any reason, the structure of the group changes instantly. Without a male to fertilize eggs, reproduction would halt, threatening the long-term survival of the colony. Yet angelfish have evolved a remarkable solution to this problem, one that relies on a biological phenomenon known as protogynous hermaphroditism. In this system, individuals begin life as females, possessing reproductive organs designed to produce eggs. Under certain conditions, however, the largest or most dominant female within the group can transform into a fully functional male. This transformation does not require generations of evolution or complex mating rituals. Instead, it unfolds within the body of a single fish, driven by hormonal and genetic mechanisms that respond to changes in the social environment. Within the angelfish group, the loss of the dominant male triggers subtle behavioral shifts almost immediately. The remaining females continue feeding, but their interactions become slightly more competitive as the hierarchy reorganizes. Among them, the largest female begins to display behaviors that were previously reserved for the male. She patrols the territory more assertively and begins positioning herself closer to the center of the group. These behavioral changes represent the first visible signs of a transformation already beginning inside her body. The trigger for this process originates in the fish's endocrine system. Hormones circulating within the bloodstream respond to environmental and social cues, altering the activity of genes that regulate reproductive organs and secondary sexual characteristics. When the absence of a male becomes clear, hormonal signals suppress the production of female reproductive hormones while activating pathways associated with male development. This shift initiates a cascade of physiological changes that gradually reshape the fish's anatomy and behavior. Inside the gonads, tissues previously dedicated to producing eggs begin reorganizing into structures capable of producing sperm. Cells that once matured into ova are replaced by spermatogenic cells, altering the reproductive capacity of the fish. At the same time, external features begin to change as well. Coloration intensifies, often adopting patterns characteristic of male angelfish in the species. Body shape may shift subtly, and the fish's behavior grows more territorial and assertive as it assumes its new role within the group. The entire transformation can occur in a surprisingly short period of time. Within days, the largest female may complete the transition and become the new dominant male. Once the change is finished, the fish functions biologically and behaviorally as a male, capable of fertilizing eggs produced by the remaining females. The colony continues its reproductive cycle with minimal interruption, demonstrating an extraordinary example of biological flexibility shaped by evolutionary pressures within the reef environment. This rapid gender shift reflects the importance of maintaining reproductive continuity in ecosystems where mortality rates can be high. Coral reefs host an immense diversity of predators, and even the most vigilant species cannot guarantee survival of every individual. By allowing a female to assume the male role when necessary, angelfish ensure that the group can continue reproducing even after the loss of its leader. The system also maximizes reproductive efficiency because the largest individuals typically become males, enabling them to defend territory and fertilize eggs from multiple females. From an ecological perspective, the strategy aligns with the resource dynamics of the reef. Larger fish often possess greater energy reserves and stronger swimming capabilities, allowing them to patrol territories more effectively. By transforming the largest female into the new male, the group preserves a social structure in which the strongest individual performs the role requiring the most defense and display. This arrangement balances reproductive potential with the physical demands of maintaining territory in a competitive environment. The reef itself provides constant reminders of why such adaptability is necessary. Predatory fish glide through the coral corridors, while small invertebrates cling to surfaces where currents deliver a steady supply of nutrients. The angelfish group continues its daily routine among these shifting dangers, feeding on sponges and algae that grow along the reef's surface. Their beak-like mouths allow them to nip carefully at these organisms without damaging the surrounding coral structure. As the newly transformed male settles into his role, the dynamics of the group stabilize once again. The other females follow his movements as they explore the territory, maintaining a loose formation that provides both feeding opportunities and a measure of safety in numbers. The male resumes the patrol patterns once performed by his predecessor, swimming along the edges of the territory and occasionally chasing away intruding fish that venture too close. During the breeding season, the male leads the females into open water above the reef where spawning occurs. Eggs and sperm are released simultaneously into the water column, where fertilization takes place externally. The fertilized eggs drift with ocean currents until they hatch into tiny larvae that begin their own journey through the planktonic stage of life. Many will not survive the hazards of open water, but those that do eventually settle onto reefs, where they grow into juveniles and join the complex social systems that define angelfish communities. The capacity for rapid gender transformation demonstrates how evolution has equipped reef species with solutions tailored to their specific environmental challenges. In habitats where population density is high and predation pressure remains constant, reproductive flexibility becomes a powerful advantage. Angelfish embody this principle through a system that ensures no group remains without a breeding male for long. While the process may seem extraordinary from a human perspective, within the reef ecosystem, it represents a practical response to unpredictable mortality. Life on the reef operates through constant adjustments to environmental and social conditions. Species that can respond quickly to these changes gain an advantage in maintaining stable populations across generations. As currents sweep gently across the coral landscape, the angelfish group continues its quiet patrol through the reef. Sunlight flashes across their scales, while the new male glides confidently at the center of the formation. The transformation that occurred within his body remains invisible to the casual observer, yet it reflects a remarkable biological mechanism that preserves the continuity of life within this intricate marine ecosystem. In the crowded world of coral reefs, where countless species compete for space and resources, adaptability becomes a defining trait of survival. The angelfish demonstrates that evolution does not always rely on physical weapons or defensive armor. Sometimes the most powerful adaptation lies within the flexibility of the organism itself, a genetic and hormonal system capable of reshaping identity to ensure that life continues even when the unexpected disrupts the delicate balance of the reef. Far from the shelter of coastal reefs and kelp forests, the open ocean stretches outward in immense, shifting plains of water, where currents carve invisible highways through the deep. The surface often appears calm, but beneath it lies a constantly moving architecture of temperature layers, migrating prey, and predators that roam vast distances across entire oceans. Here, where visibility can extend for kilometers and the scale of the ecosystem dwarfs nearly every coastal environment, one hunter has evolved an advantage that goes beyond teeth, speed, or strength. Among the most formidable predators in the sea, the orca dominates its environment not merely through physical power, but through a trait far rarer in the animal kingdom: culture. The ocean surrounding a hunting pod of orcas may appear quiet at first glance. Swells rise and fall slowly under pale northern skies, while distant seabirds glide across the horizon. Beneath the surface, however, a coordinated group of black and white shapes moves with extraordinary precision. Each individual maintains a carefully measured distance from the others, adjusting position subtly as they travel through the water. Their movement appears effortless, yet it reflects a complex social system that shapes every aspect of their lives, from hunting to communication and migration. Unlike most predators, whose behavior relies primarily on instinct encoded in genetic patterns, orcas possess a brain capable of storing and transmitting learned knowledge across generations. Their brains are among the largest and most complex of any marine mammal, containing expanded regions associated with social cognition, memory, and communication. This neurological capacity allows orca populations to develop distinct behavioral traditions that function much like cultures within human societies. These traditions vary dramatically between different pods. In one region of the ocean, a group of orcas may specialize in hunting fish, targeting species such as salmon that migrate along coastal waterways. In another region, a different population focuses almost exclusively on marine mammals, pursuing seals, sea lions, or even whales across the open sea. The remarkable aspect of this variation lies in how these hunting strategies are maintained. The behaviors are not merely inherited through genetics, but learned through observation and practice within family groups. Within an orca pod, young calves spend years learning the skills necessary to survive. From the moment they are born, they remain close to their mothers and other relatives, observing every movement and interaction. When adults hunt, the calves watch carefully as the group coordinates its actions. They learn how to position themselves during a pursuit, how to interpret the vocal signals exchanged between pod members, and how to recognize the subtle cues that indicate when prey has been located. Communication plays a central role in this learning process. Orcas produce a complex array of whistles, clicks, and pulsed calls that travel efficiently through water. These sounds serve multiple purposes, including navigation, prey detection, and social interaction. Each pod develops its own unique repertoire of calls, often described as dialects because they differ noticeably between populations. Just as human languages vary across cultures, the acoustic signatures of orca pods reflect social identity and lineage. These dialects are passed down through generations, forming an auditory map of social belonging. When individuals encounter unfamiliar calls, they can recognize that the source originates from a different pod or population. This ability helps maintain the integrity of social groups that rely on shared knowledge and coordinated behavior to hunt effectively in the vast ocean. One of the most striking examples of culturally transmitted hunting behavior occurs in the icy waters of polar regions, where seals rest on floating platforms of sea ice. In these environments, a single seal perched on a stable ice floe might appear safe from predators below. Yet pods of orcas have developed a technique known as wave washing, a coordinated strategy designed specifically to dislodge prey from ice. The hunt begins when the pod locates a seal resting on a piece of ice. Rather than attacking individually, the orcas position themselves in a line several meters from the floe. With precise timing, they swim rapidly toward the ice while rising toward the surface. As their massive bodies surge upward, they generate a powerful wave that travels directly toward the floating platform. When executed correctly, the wave breaks across the ice and washes the seal into the water, where the pod quickly surrounds it. This strategy requires extraordinary coordination. Each orca must match the speed and direction of the others to produce a wave strong enough to destabilize the ice flow. Young individuals spend years practicing the technique under the guidance of experienced hunters. During early attempts, they may observe from the edges of the formation, while adults demonstrate the proper timing and positioning required for success. The technique also demonstrates how knowledge spreads within a pod. Once a particular hunting strategy proves effective, it becomes part of the group's behavioral repertoire. Calves born into the pod grow up observing the behavior repeatedly until it becomes second nature. Over time, the strategy evolves through subtle refinements as individuals experiment with new variations or adapt to changes in the environment. These cultural traditions extend beyond hunting techniques to include migration routes and seasonal movements. Orca pods often follow specific paths through the ocean that align with the movements of their preferred prey species. These routes can span thousands of kilometers, connecting feeding grounds, breeding areas, and migratory corridors used by prey animals. The knowledge of these routes resides within the memory of older individuals, particularly matriarchs, who lead the pod through complex ocean landscapes. The role of matriarchs within orca societies reflects another dimension of their cultural organization. Female orcas can live for many decades, often remaining active within the pod long after they stop reproducing. During this extended lifespan, they accumulate vast knowledge about the environment, including where to find food during periods of scarcity and how to navigate shifting ocean conditions. Younger members rely on this experience to guide their own survival, demonstrating how the wisdom of elders contributes directly to the resilience of the group. Predator and prey relationships within this ecosystem further highlight the sophistication of orca hunting strategies. When targeting schools of fish, orcas may employ herding tactics that concentrate prey into dense clusters before striking. In other situations, they use echolocation clicks to track the movements of animals hidden beneath the surface. These clicks produce sound waves that bounce off objects in the water, returning echoes that reveal the size, shape, and location of potential prey. The ability to interpret these echoes requires advanced neural processing, allowing the orca to construct a three-dimensional acoustic map of its surroundings. Combined with visual cues and social communication, this sensory system gives the pod an extraordinary awareness of its environment. Each individual contributes to the collective perception of the group, sharing information through vocal signals that coordinate their movements during hunts. Occasionally, the prey pursued by orcas rivals them in size and strength. In certain regions, pods have been observed attacking large whales, including species many times their mass. These encounters involve complex strategies in which the pod isolates weaker individuals and uses repeated strikes to exhaust them. Such hunts demonstrate the full extent of the orca's cooperative abilities, where each member performs specific roles within a coordinated attack. Yet the cultural dimension of these behaviors remains the defining feature of orca ecology. Different populations exhibit entirely distinct hunting traditions, even when they inhabit similar environments. Some pods ignore prey species that others target regularly, suggesting that learned preferences guide their choices. This variation illustrates how culture can shape ecological roles within the same species. The presence of culturally distinct populations has profound implications for conservation. Protecting orcas requires preserving not only the animals themselves, but also the knowledge embedded within their social structures. When a pod loses experienced individuals, the cultural memory of migration routes or hunting strategies may vanish with them. The survival of these traditions depends on stable family groups where learning can continue across generations. As the ocean darkens toward evening, the pod continues its journey through open water. Their dorsal fins cut clean lines across the surface before disappearing again beneath the waves. Each movement reflects a blend of instinct, experience, and social learning that defines their existence as cultural predators. Within the immense scale of the ocean, the orca's greatest adaptation is not merely its size or strength, but the capacity of its mind. Through communication, cooperation, and the transmission of knowledge, these animals transform hunting into a collective art refined over generations. The strategies they employ, from wave-washing seals to coordinating attacks on large prey, represent traditions preserved through memory and teaching rather than genetic programming alone. In this sense, the orca occupies a unique position among marine predators. It is not only an apex hunter, but also a carrier of culture, navigating the ocean with intelligence shaped by shared experience. The waves roll steadily across the open sea, and beneath them, the pod moves as a living archive of knowledge, each individual contributing to a legacy of survival written not in genes alone, but in the learned behavior of one of the ocean's most sophisticated minds. Along the margins of tropical coral reefs where sunlight penetrates the upper layers of the ocean, the reef appears almost impossibly alive. Hard coral structures rise like towers and buttresses across the seabed, their calcium skeletons built slowly by countless generations of tiny coral polyps. Algae grows across exposed surfaces, while sponges expand through shaded crevices in a riot of color. Fish move constantly through this architecture, forming shifting schools that flash silver and yellow as they pass through columns of light. Yet beneath the surface beauty of this ecosystem lies an intricate network of predators, ambush specialists, scavengers, and opportunistic hunters that keep the reef in ecological balance. Among the most unusual of these hunters is an animal whose anatomy seems almost engineered to solve one of the most difficult problems in aquatic predation. Within the narrow tunnels and cavities of the coral structure lives the Moray eel, a predator that spends much of its life hidden from open view. At first glance, only its head protrudes from a dark crevice, jaws opening and closing slowly as water passes across the gills. The eel's elongated body remains concealed deep within the reef structure, coiled like a living rope inside the protective labyrinth of coral chambers. The opening and closing of its mouth often appears threatening to casual observers, yet this behavior is simply the eel's method of breathing. Unlike many fish that pump water across their gills through constant swimming, the moray must repeatedly open its mouth to draw oxygen-rich water into the throat. From this concealed vantage point, the eel watches the surrounding reef. Its vision is modest compared to the sharp eyesight of many reef fish, yet its sense of smell is highly refined. Chemical traces drifting through the water reveal the presence of potential prey long before the eel sees movement. A small fish swimming through the coral maze leaves behind a faint trail of scent molecules that linger in the current. For the moray, these chemical signals function like invisible pathways through the reef, guiding the predator toward possible meals hidden among the coral branches. The reef itself provides both shelter and hunting grounds for such a predator. Countless small fish species depend on the coral's complex structure for protection from open water hunters. They weave through narrow spaces between coral heads, darting into crevices at the slightest sign of danger. For most predators, pursuing prey through such tight spaces would be difficult or even impossible. The moray eel, however, possesses a body perfectly adapted for navigating this environment. Its long, flexible form allows it to slip through gaps that larger predators cannot enter, turning the reef's defensive architecture into a hunting arena. When the eel decides to strike, its movement is sudden and precise. The head lunges forward from the crevice with remarkable speed, jaws snapping shut around the prey before the victim has time to escape. The front jaws of the moray eel are armed with sharp, backward curved teeth designed to grip slippery fish and prevent them from wriggling free. At this moment, the eel faces a challenge shared by many predators that hunt in confined spaces. Unlike animals with powerful limbs or suction-based feeding mechanisms, the eel must pull its prey into its throat while holding it firmly with its jaws. For most fish, this would require repeated adjustments of the bite, risking the loss of the struggling prey. Evolution has solved this problem in the moray eel through one of the most remarkable anatomical adaptations found among vertebrates. Hidden behind the primary jaws lies a second set of jaws located within the throat. These structures, known as pharyngeal jaws, exist in many fish species in a reduced form used primarily for processing food once it has already entered the mouth. In the moray eel, however, the pharyngeal jaws have evolved into a fully functional grasping apparatus capable of extending forward toward the mouth. When the eel bites its prey with the front jaws, the pharyngeal jaws activate almost instantly. Driven by specialized muscles, the inner jaws slide forward from deep within the throat until they reach the captured prey. The teeth of the pharyngeal jaws clamp onto the victim, and pull it backward into the esophagus, while the front jaws release and reset for the next bite. This sequence unfolds with remarkable speed, transforming the eel's throat into an internal conveyor system that transports prey deeper into the digestive tract. The mechanics of this feeding system appear almost alien when observed closely. As the eel captures its prey, the second set of jaws emerges from the throat like a mechanical arm, grasping the prey before retracting again into the body. The process eliminates the need for suction feeding or complex head movements, allowing the eel to swallow prey efficiently, even while confined within the narrow spaces of coral cavities. This design offers enormous advantages in the reef environment. Small fish that attempt to hide within the coral structure may find themselves cornered by an eel capable of striking and consuming prey without needing to reposition its body. The eel can remain partially hidden while feeding, reducing its exposure to larger predators that patrol the reef's outer edges. Moray eels occupy an important ecological role within the coral reef community. By preying on smaller fish and crustaceans, they help regulate populations that might otherwise grow excessively and disrupt the balance of the ecosystem. Their presence ensures that no single prey species dominates the reef's intricate food web. The eel itself must also remain alert, as larger predators such as sharks occasionally hunt within the reef environment and may attempt to capture an eel if the opportunity arises. Interestingly, moray eels sometimes engage in cooperative hunting interactions with other predators. In certain reef systems, groupers have been observed signaling to morays through specific body movements that encourage the eel to leave its crevice and join a hunt. The two predators then move through the reef together, exploiting each other's strengths. The grouper patrols the open water around coral heads, while the moray investigates narrow crevices that the larger fish cannot access. When prey attempts to escape one predator, it often moves directly into the reach of the other. Such interactions highlight the complexity of predator relationships within coral reef ecosystems. Cooperation between species can improve hunting success for both participants, illustrating how ecological pressures sometimes favor unexpected alliances. The moray eel's flexible body and specialized jaws allow it to exploit spaces that remain inaccessible to other predators, making it a valuable partner in these opportunistic hunts. As night approaches, the behavior of many reef organisms changes dramatically. Diurnal fish retreat into coral shelters, while nocturnal species emerge from hiding to begin their own feeding activities. Moray eels often become more active during these hours, leaving their daytime crevices to patrol the reef more openly. Their sense of smell becomes especially valuable in low light conditions, guiding them toward prey hidden within the coral labyrinth. Despite their formidable hunting equipment, moray eels rarely pose a threat to larger animals unless provoked. Most encounters with divers occur when an eel remains within its crevice, observing its surroundings with cautious curiosity. The repeated opening and closing of its mouth continues as water passes across the gills, creating the illusion of aggression, even though the animal simply breathes. The evolutionary history of the moray eel stretches back millions of years, and the basic design of its feeding apparatus has remained remarkably stable across that time. The pharyngeal jaws represent such an effective solution to the problem of capturing prey in confined spaces that the fundamental mechanism has changed very little through evolutionary history. In this sense, the moray eel stands as a testament to the power of natural selection to refine biological systems until they achieve an efficiency that requires no further modification. As currents sweep gently across the reef and schools of fish move through the coral structures, the moray eel waits patiently within its rocky refuge. The reef remains a place of constant movement and interaction, where survival depends on specialized adaptations suited to an environment crowded with competitors and predators. Among these adaptations, the moray eel's internal jaws represent one of the most striking examples of evolutionary innovation. Within the narrow chambers of coral stone, where light fades into shadow and prey passes unknowingly through hidden corridors, the eel's alien jaw mechanism continues its silent work. The predator does not rely on speed alone or brute force, but on a design that transforms the simple act of swallowing into a precisely engineered system. In the living architecture of the reef, the moray eel remains a quiet but formidable presence, a hunter shaped by millions of years of evolution, to thrive within the maze of coral that defines one of the ocean's most intricate ecosystems.